there it is, right next to it.
Something.
Great.
Yeah, just like, you know,
Where's the
60 meters.
Which microphone of these three is the
Silver one.
that one?
Yeah.
Okay.
Yeah.
But whenever you talk, you're supposed to look directly up.
No, I'm not. No, that's weird. Okay. Okay. All right. Hello. Okay. All right. So, good morning. Is it morning? It is here. This is Rachel Lauer from the University of Calgary. I am just here to get us started because the first exciting part of this dive will be a heat flow measurement. We are back to a similar location to where we were yesterday. We were on one of two carbonate mounds. They were just massive biogenic limestone mounds that are formed on top of these sediments. These have been visited in 2014 and written about in a publication in 2017, but only a few of the sites that we're going to visit had been previously seen with human eyes. So we are in a site that has not been seen, but has been mapped. We mapped some of this overnight and this morning, so we have a really good detailed map of these structures, and we're on the east side of one that we are going to climb up on top of throughout the course of this dive. And there are, in previous work, identified these massive beds of mussels and other chemosynthetic animals that are living on top of this mound. Also, lots of methane hydrite. So, but before we do that, we are looking for a soft place to conduct a heat flow measurement. We need at least 60 centimeters of sediment to be able to conduct that measurement safely. And I think anywhere you guys are, I mean, your guess is as good as mine where there's 60 centimeters of soft sediment. You want to try anywhere?
We can do it here if you want, but
Yeah.
we're not quite to waypoint one.
Yeah, I mean, waypoint one is
New waypoint for you.
where you're starting the transect.
Good morning, thanks a lot.
There's a rock. Don't want that.
What's the current? How about you just tell me when you want to stop?
Yeah, I mean, we want to do a couple measurements approaching the mound. So whatever angle that takes, if you want to first get to waypoint one, that's fine.
I mean, it makes sense to me, and that way you know they're all
Yep, in a line.
on the line or whatever. But
Cool.
to us. Will you check the heat flow probe pressure? Make sure it's 37% on the valve page. And the Niskin's 27.
And maybe, is it possible to put the heat flow probe in the corner here?
You could put it the whole screen.
No, we can't do that.
Okay.
Okay, never mind. No one wants to see that.
I mean, it would just be your screen. So.
Oh, no, it's okay.
Okay.
I just meant in the screen so that, you know, so people can see the important science that's happening.
Oh, on the out.
Yes,
Yeah. I thought you meant
yeah.
yeah.
Like, now, that would pretty much ruin our viewership if we
Yeah,
just show
37?
that.
About 6.
There you go.
25.
Yep, set it to 37.
So,
I've tried clicking save settings.
Yeah, it doesn't work. You can try it again if you want. You go to publish, and then
All right, so if you missed yesterday's dive,
3, right?
we were on a mound to the west of us that was just covered in methane seeps. Oh, what is this little, is that a sea cucumber? A spiky looking sea cucumber? Just sailed past one. And did a bit more mapping overnight and are going to spend today on a slightly larger, but similarly formed mound of carbonates that's both chemosynthetic and biogenic carbonates. I mean, it's if you look in closely in some of the what we called rocks, they are basically just enormous mats of life that existed at some previous time, maybe millions of years ago, that have now become glued together with lots of carbonate over time, lithified, and just created more and more elevation above the seafloor.
Very bottom. Very bottom. No, very bottom to the right. Very bottom there. Yeah, I think it's like a permissions problem. We got to figure out where that file is and why it's not allowing us to write to it. But, what's that? Oh, Excalibur? Yeah, we'll do Excalibur. You want Excalibur, right?
Yes, to Excalibur. Yeah, and if we can, like we were talking about, the poke it and hold it, and then
Well, like a
yeah,
hold
like
it
an
still.
anchor,
Sure. Yeah, we can do that.
just to
The
maybe it'll
current
be
is
easier
also-
to deal with. Is it better?
It's a lot better today. So far.
Okay, that's good. Yeah, we, in addition to having some weather at the surface, we also yesterday had a really strong current that was making it really difficult to move in any direction except to the northwest. So the sub spent the day and the pilot spent the day really fighting that current, trying to achieve our dive goals. So hopefully today it's a little bit easier.
It's probably just in my head, but it seems like the more the seas come up, the less they come
Oh, interesting. Like all the energy is like somehow in the near surface now
It's
instead
probably
of
just in my head, but
Yeah.
it really does seem like that a lot.
interesting. Yeah, it sounds like we're gonna get bigger seas over the next couple of days, so we're all preparing for that. And we're heading right now to our first waypoint, and the reason I'm on the mic instead of one of the biologists is that what we're going to do first is collect a heat flow measurement. We did collect one yesterday between Mound 1 and Mound 2, and I didn't name them, but that's what they're called. And these are these massive carbonate mounds at the seafloor, and they're sitting on top of the Barbados accretionary prism, which is, I believe, the largest accretionary prism on the planet. It's about, in this region, it's up to like 8 to 10 kilometers of sediment that has been scraped off of the South American plate onto the Caribbean plate. Oh, cool.
Interesting.
What's that? Besides sargassum and some,
What coral?
yeah,
Rocks.
rocks. Yeah, thanks, guys. That's.
You think those are authentic rocks?
I mean, are they authentic rocks or are they fake
They look
rocks?
authentic.
I
They
think
look-
they're authentic.
I think they're authentic.
Okay, and also autogenic.
What is autogenic?
I just made it up. No, it means. Do you want to explain what it means?
Authentic or
Autogenic.
You can pick.
It means that it's formed by processes, chemical processes.
Biogeochemical.
Yeah, biogeochemical processes.
So that would be like creatures that make shells.
Yeah.
And shells.
Yeah, exactly.
Combination of-
Yeah, so that's, I mean, again, not I'm a geo person, but, you know, like biology, if I asked a shark person about microbes, they're probably not going to know the answer, so I'm kind of winging it here, but the structure and the platform that creates these mounds is both chemical and biologic, because the chemistry creates the carbonate and interactions with bacteria and chemistry And then that brings all of the life to that spot, the tube worms and the mussels. And as those seeps die out and their lives die out, they create the platform for the next layer of carbonates. So, yeah, these structures.
That's an autogenic rock.
Yep, an autogenic situation. Rock.
So kind of like those ones we saw.
Not an authentic rock, but an autogenic rock. Yes.
Well, I mean, you could have an authentic autogenic rock.
You could,
Absolutely,
yes.
or a fake one.
But yesterday we saw a lot of these rocks that were like worm tubes.
Yeah.
That kind of thing.
Yeah, exactly. So the rocks, if you zoom in on the rocks, which most people don't, what you'll see is, the past inhabitants now lithified as a rock. So you can see the tube worms, you can see the mussels. Obviously, there's some sediment and time that have turned them into rocks. But yeah, it's cool. And how tall these are, I'm not sure how tall they are from the seabed, like 300 meters? I'm not sure. Do you remember, Rob, how tall they are?
Domes
The domes.
in that neighborhood.
Yeah, hundreds of meters. So lots of times these have been being built, so millions of years. And these vents, they can stay active for thousands of years, millions of years. But it's more likely that they migrate around and create similar structures in this area where you have lots of sediment and lots of pressure at depth that's squeezing these waters that are rich in methane and carbon dioxide and other things associated with hydrocarbons. So I think this looks like a nice spot for us. Are we at our cool? All right,
So
we're gonna.
stop in a second.
So first, before we use our heat flow probe, which is mounted onto the side of the sub with a hydraulic ram that allows it to move perfectly vertically into the sediments. In other cruises, we've used the articulating arm, which is super difficult to do in terms of trying to get it perfectly vertical. This way we know it's going vertically in. And before we do that, make sure we're not going to damage the probe. We're going to poke around with what was named Excalibur by our pilots to make sure that we have enough soft sediment, at least in this area. We might locally encounter a rock, which we did yesterday. We'll have to move to find that soft sediment, but this gives us a sense of the thickness to make sure that we're not in a thinly sedimented
Can I
area.
grab it and then re-grab?
You can grab whatever you need to.
Because I won't be able to grab it at all. But
They just want to see that it's soft, right? So you can probably do a side grab. And then you're just gonna keep it in the mud once you.
that will be our spike.
Maybe keep your claw on it, right? Okay. Hand, claw, I don't.
My favorite was a guy
I feel
called
good
it the
about this.
pinchers.
The pinchers.
Yeah.
I mean, they are pinchers, right?
Grab that with your pinchers.
Was he Southern?
Ready?
Yeah. Where from?
Somewhere
Somewhere that they say pinchers.
Probably some
Yeah.
of those crawfish, I'm guessing.
Yep, crawfish.
How close you want me to? What position?
That's
you want me to
it's
test?
pretty much anywhere. We're just trying to get a sense of the sediment
Okay.
thickness broadly in this area.
It's thick. It's gonna be good, Rachel.
Yeah.
Yeah?
Looks soft. Keep going. Keep going. Can we keep going? All right. That looks good. Oh yeah. Okay.
You want me to leave it here?
Nope.
as-
If just hang on to it if we can as an anchor.
freeze your pinchers.
Not for now.
Oh, okay. Yeah. Oh, thank you. Cool.
Okay.
All right, so now that we've made sure that there's enough sediment, we're gonna go ahead and try a heat flow measurement. Did you get to fire the heat pulse on the way down or no?
Yeah.
You did? Looks good? Okay. All right. Little urchin we can zoom in on so that everyone's not bored while we do this. Let's go ahead with the heat flow probe.
Going down.
Cute.
All the way, right?
Yep. All the way. Yes. Awesome.
Your heat flowing.
We're heat flowing. So what's behind? What are those little tubes behind?
I'm trying to figure
Yeah.
out.
Just trying to figure out what's going on behind the urchin.
That's maximum, unfortunately.
What is it? It is? No, that's fine. I don't know if zooming would help. It's like a tube looking thingy.
Very regular.
Huh.
Maybe there's more.
Yeah.
Closer.
So the way this tool works, it is titanium housing and a titanium lance that we just pushed into the seafloor. It's about 60 centimeters long, and then within the lance itself are evenly spaced thermistors, which are measuring temperature. And the first part of the measurement, for 7 minutes, so go get a drink or coffee if you need one, is frictional heating. So as you're pushing it in, that friction is going to cause temperature to rise. And then after about 7 minutes, you can get a sense of what the geothermal gradient is. And that's just how much is temperature increasing below the seafloor. And that's your geothermal gradient. And then after 7 minutes, we will fire a heat pulse, which puts a known amount of heat along that lance. And the way that that decays over the next 7 minutes, the second 7 minutes, gives us an estimate of thermal conductivity. And thermal conductivity is just like how easily heat moves through a sediment. So some sediments are resistive to heat transport, some are very conductive. So you get a sense of how that is changing beneath the seafloor. And then with a little bit of math and multiplying those two together, you end up with what we care about, which is heat flow. And in this area, it's possible there's some weird stuff happening because of the presence of large amounts of methane hydrate, which obviously we're not seeing. That guy's kind of cool. Is that the current direction? The way that guy's going? Yeah.
It's
Okay.
not too great right now.
No.
What are these things we're looking at? The
I don't-
triangular things.
I think these are just shell fragments, aren't they?
Foreign.
Or they look really sharp.
Yeah, you wouldn't want to step on that.
No, you don't want to step on that. I assume they're shell
Yeah,
fragments?
they're shells of theropods.
Pteropods. Okay. Yeah, so we're right next to a structure, a mound where there's lots of carbonate and animals, and this whole area has been littered with little fractured shells and the body parts of other things that are no longer with us. Oh, there's a little, what is that? Sea cucumber or something smaller
Where are you looking?
over the left?
Floating?
Yeah.
You mean floating?
Oh, that's small. Okay. I don't have the lasers. It's a teeny
Worm?
thing.
Molecule resuspended?
Polychaete. Okay.
There's a
Yeah. Okay.
Oh. Everything here looks cool when you zoom in on it. Look at that.
You're killing me. You're killing me, little guy.
Yeah, so these thermal measurements are the original technology was developed in oil and gas industry to understand like the thermal maturity of hydrocarbons, and we use it now for primarily vents to understand the pattern of fluid movement and fluid exchange between the ocean and the underlying crust. So in places where you have vents, here we're looking at cold seeps, but sort of the cousins to the cold seeps are the vents, where you have water moving in seamounts or outcrops or extinct little volcanoes, going into the crust, warming up, and then coming out somewhere else. And if you were to do a transect between two of these little outcrops or little volcanoes, extinct volcanoes, so they're basalt, you would see the heat flow rising as you go towards the discharging outcrop, where water is coming back into the ocean after getting warmed up, and heat flow would be really low where the water is going in, because the bottom water is very cold relative to the crust. So it gives you a sense of those patterns that exist between the ocean and the ocean crust. And it also provides some quantitative information about how we should be thinking about establishing protected areas, because these areas where fluids are coming out tend to be biodiversity hotspots. And if we are interested in protecting those regions from things like deep sea mining or other activities that could harm the biology that we care so much about, we would want to also protect the areas where the fluids are going into the crust. So this is a great tool for that. How are we going as far as the measurement?
We're approaching the heat flows.
Okay. So that's how biologists and geophysicists end up working together, is the tools that we use end up being really helpful in thinking about the fluid flow that's happening that we can't see, but we can measure using tools like heat flow. And in this particular environment where we have a massive accretionary prism, a lot of the flow is happening from much warmer parts of the accretionary prism, so kilometers below where we are. And these active seep sites may have temperatures that are slightly above background, so it's hopeful that we can collect sort of the signature of those cold seeps. Or later in the cruise, when we are conducting a similar transect across another structure, this time a mud volcano, which are prevalent all over the seafloor in Trinidad and Tobago's waters and all over southern Trinidad. I think there's like over 20 mud volcanoes on land as well. How does it look?
It's the same pattern.
Yeah. Yeah. Okay. Cool. All right, so the heat pulse is fired, and we're gonna wait until the spike associated with that heating declines, and that takes about another 6 minutes, and then we will move to our next location. All right, so those of you just joining us, we are not going to stand here all day looking at this sea urchin. We are conducting a heat flow measurement, which takes about 15 minutes. So we're trying to give you something cute to look at while we're doing that. There will be lots of biology later in the dive. We are heading to a carbonate mound that is just covered in all sorts of seep creatures. So lots of mussels, and we saw yesterday some skates, lots of shrimp and gastropods and other creatures that you will see later in this dive. But right now, we're hanging out and looking at this urchin while we finish our measurements. Yeah, so I think that tube is like one of those things at the bottom there. Maybe? I don't know.
The mountain?
Yeah. Oh. No. Yeah. Hanging out and being, yeah. Yeah, maybe this one's moving too much. Oh, there's a sea star or a brittle star. Cool.
Brittle star.
Brittle star.
Thin arms, brittle star.
What's that?
Those thin arms, the
yeah.
Niskin thin arms,
Long
or
skinny
brittle?
arms. Yeah.
Brittle, you know, brittle.
Yeah, we saw a couple beautiful corals yesterday. Lots of brittle stars. And he's I've never seen one move like that. That's kind of awesome. Slowly making his way across the seafloor. How we doing? Cool.
So we have the front seepage, which is out dive. You see Rachel there, and she's really kind of directing the pilots on this.
So for our next location, we're going to get a little bit closer to this
Very skilled
massive carbonate mound. Collect another measurement before we start going up the slope and heading towards the top,
And right here we have
where we expect to encounter
one of our
something similar landscape to what we had yesterday. Except I think
animal
well, we'll see, but there is an expectation of even more methane concentrated in this particular part of the seafloor. So they actually even have
she's in control of the entire
higher
dive
concentration
operation,
of
and,
animals
a wealth
as
of
well.
knowledge we're quite lucky to have her. Anna, thanks.
And here we have our artist at sea, Marco.
Hi, everyone.
I don't know if they can take
And Kyle, our resident Bajan, respect.
who just had his first speech.
Woohoo! Congrats to Kyle.
Congrats, man.
Okay, so that's mission control. We're just gonna take a quick look.
Right, so a lot of the chatter you just heard, we do a lot of ship to shores. So I think one of the schools, we're doing a ship to shore where we have a tour of the ship. So they were just in the control room introducing the kids to pilots and scientists, which is awesome. We got to create the next generation of marine biologists and marine geophysicists. So,
Right on.
all right, how we doing? All right, so we're good to
Good?
extract the probe and pew pew pew. Oh no, that's a laser noise. All right, I don't know. I don't know what the noise is for the heat flow probe. I'll think about it. Come up with something better.
Womp womp. All right.
Good morning.
I don't think
morning.
you
How's-
have.
Good. How are you?
Pretty well. Looking forward to today.
Yeah.
Some cool stuff.
Yeah, we will. Not for a bit. So this time we're using Excalibur as an anchor, which I think yesterday it seemed like a good idea when there's that much current, right? Because it kind of holds us in position, but.
It was so calm, you can see the sediment.
Yeah, I know, it's not, it's
Why
total-
not
but
pre-
why
why
not,
not
right?
prevent?
Why not, yeah.
You
Um,
grab it on the
So
scalp.
since we're poking it, we might as well use it, right? So we're ready to head towards that guy, and if we could, we're gonna get, as long as we have sediment cover, we'll do one more heat flow before we get to the fun stuff. Not to say heat flow is not fun.
Rachel,
Yes.
how far do you need the next?
We're
Close or far?
I mean, probably 100 meters at least, yeah. So if we could, thank you.
No, that's fine, just trying to figure out transects, other
Yeah.
transects. So we'll do another hit flow, then we'll go transect.
Yep, yeah, exactly. I mean, hopefully we see something on the way.
Then we back, stop, no
Yeah.
hit flow,
Yeah.
just the rest.
Yes, exactly.
And collection, authentic rocks.
Authentic.
Okay,
So we're gonna pull up Excalibur, keep him in your arm, claw, hand, and we'll use it the same way at the next site, and hopefully we can do another one in 100 meters before we get to the places where there's no sediments. Looks like you're ready to fight somebody.
Okay. Yeah.
So we're at waypoint 1, right?
We are.
Okay, so that thing's not up to date. Okay. Got it.
I think if they zoomed into that, you were too low.
Oh, really? Okay.
Yeah, we can start off WrestleMania.
Camera.
Hmm?
Camera. Street it. We keep the vessel behind you or the side of you?
We'll try it behind us for them to see how it goes.
Hanging like this. Huh?
Yeah.
What's the water?
Well, that's all called separate.
Okay. Cool. Same as yesterday.
Bridge, new waypoint.
Okay, wait.
Just gonna wait for the vessel to start.
Yeah, sure. How far or how close do we have to work the ship? Is it okay?
Yeah, this distance fine. I just don't want to get too far out and then we'll get this tight.
Oh, yeah. Pulling. Yep. Okay, so if you're just joining us, we are still offshore Trinidad and Tobago. Today we are diving in a similar location to yesterday. Yesterday we spent the dive on a carbonate mound at the seafloor that's a couple hundred meters high and is the result of millions of years of chemosynthetic and biogenic processes that together lead to the development of what looks like a little mountain of carbonate at the seafloor. And that is the result of us being on a massive accretionary prism, which is all of that sediment that was scraped off South American plate onto the Caribbean plate over millions of years has led to this giant pile of sediment. And at the base of those sediments are all the ingredients for the development of massive hydrocarbon resources, which is an important part of Trinidad and Tobago's economy. But also in combination with that are methane and CO2. And that methane, under that pressurized condition, and squishing and consolidation and deformation of those sediments deep at the base of this pile, is creating enough pressure and finding fractures and faults that are pathways for that pressurized fluid and gas to make its way all the way to the seafloor. And so that gas, when it makes its way to the seafloor, is assisted by bacteria that, in combination with other microbes, are taking that methane and sulfur elements and turning them into carbonates. And that carbonate then is providing a hard surface for an attachment place for lots of these other critters.
Can move it.
And over time, that methane seep develops into a large community of, while it's active, of mussels and shrimp and what's the word I learned the other day? Oh wait, hold on. These are cute little things. What are they called? Eel pout.
Those are the fish.
Yeah, they're so cute.
We need to collect some.
Yeah, and those are animals that also tend to cluster on these active methane seeps, or cold seeps, they're called colloquially. And today we're going to sample some of those species that I just mentioned. I think we just want to go 100 meters from waypoint 1. Oh yeah, there it is. Cool. So yesterday, in addition to all the creatures that are attached to the seafloor, we also saw what may be a new species of octopus. New species of Rhinelodone octopus, which was very cool. And I think we've seen like 3 of those in 7 or 8 dives that we've done. How's the current? Is it as bad as?
It's okay, I think it's more.
Oh. Ships raining in.
Yeah, so when we stop for this one,
Yeah.
I think that's it's kind of heading us to see what happens.
Okay.
So 100 metres from that waypoint 4.
So now,
We're going to stop at 100 metres from waypoint 1 to do a big flow.
but you not.
Yeah, you have to follow me to the starboard, follow it towards the vessel.
So you think if I bring the vessel behind you, you will get equal less? Exactly. Like more.
Directly behind. Let's
Yeah.
try that. So when we stop at this marker, we can position the vessel.
Yeah. well, you will have to move forward a little. So I'll stop the vessel here.
Yep.
Bridge, you could come to us now.
Come into a stop.
All right, so, oh, we're starting to get onto, is that a rock or a pile of sargassum? I don't know. It's a rock. You know, you'd think with a geoscience degree I could identify a rock, but I know. I'm a bit nearsighted, especially when I'm underwater.
Although things are bigger when you're.
They're bigger. Are we already on the slope? No. Okay. A little bit? Yeah.
You want it to the 8 foot here?
Yeah. Do you want to try and get it here? We're pretty close. Yeah. Okay. So exciting times to our listeners. We are going to do another heat flow measurement. Before we do that, we're going to use Excalibur, the pokey thing, to measure the depth of the sediment and make sure we've got enough to actually use the whole probe to make a measurement. So it's made out of titanium, but if we do encounter a rock and try and ram it into the sediments, we can actually break it. So this is a test of for the safety of our instrument.
Is it slightly uphill?
I think if it's slightly uphill, sometimes to turn that way. Is that?
Turn the sub to
Yeah.
the-
Turn the sub to so the probe is on the uphill side sometimes.
It's going to land at an angle,
Okay.
and you've got potential to bend the probe.
Yeah.
I can move back to a flatter area.
No, it's not
too much of an incline.
Okay. Yeah, that's fine then. Is it hard there? No. Oh, it is. Okay. All right. Cool. All right. So it looks like we've got sufficient soft sediment to take a measurement. I think are we still powered up?
Yeah, I'm just calibrating, just waiting.
Okay, we're just gonna, first thing we do is collect a few measurements to determine the bottom water temperature, which it's about 4.7 degrees Celsius here. But that provides us a baseline for understanding how temperature is changing underneath the sediments.
Okay, ready anytime.
Okay, so we are ready. Fingers crossed. Keep going, just all the way in unless it looks like it's. Is that all the way? Okay. Rub your face.
What's wrong with my face?
I don't. We did not. No. Okay, so we're using Excalibur to kind of anchor the sub while we do this. I'll explain it again for those of you that weren't here, but the first part of this measurement is pushing that 60 centimeter lance into the seabed. Within it, it has 9 thermistors that are evenly spaced along the lance. And so first thing that happens is you get some frictional heating as a result of pushing it into the sediments. Not a lot here because this sediment is very soft. You can imagine if you're pushing it into like sand, you would expect a lot more heating. And then after that, frictional heating calms down and the temperatures stabilize after about 7 minutes. We then have our geothermal gradient, or a measure of how temperature is increasing as a function of depth below the seafloor. And then at that point, we fire a heat pulse, and that puts a calibrated amount of energy along that probe. So the temperature is going to spike, and then after 7 minutes of that temperature decay, we can use the shape of that decay to estimate the other property that we need, which is thermal conductivity. And that can also vary as with different layers in the sediment. Different types of sediments are more or less conductive. And then those two things together give us what we need to calculate heat flow. And for this part of the seafloor, it's been relatively low heat flow, but more important are how heat flow changes across the seafloor in different locations. And places where fluids are coming from really deep within the sediments, so kilometers below us, those fluids will have a higher temperature, and so higher measurements of heat flow can tell us places where fluids are, or seeps are happening at the seafloor, and that is one of the goals of this cruise. This is also a really unique area because of the hydrocarbons, because in conjunction with the oil and gas that is obviously economically important, there's a lot of methane and methane hydrate in this area that can make the heat flow signature and some of these measurements look different here than in other places, which is interesting. We know there's lots of fluid escaping at the seafloor because we wouldn't have these massive communities of animals that are chemosynthetic and rely on the sulfur and methane that are coming with those fluids for their survival. So that biology is a very good indicator of where fluid flow is happening.
Great job. Hey Tom, so you're having a little difficulty with the current down here somewhere. Just try to bring you a little more south. I put in a new waypoint for you. I'm going to try to see if the umbrella got better.
Okay, yeah. I'll put on right there.
One is the top one? I don't think that one's in.
Oh,
Yeah, look.
You want that there?
That one.
Yeah,
Because of the slope.
Looking
it's just a
forward.
curve.
Yep.
It's a camera here, isn't
Yeah, it might not.
It was-
1 and 2 might
Or is
not.
the one
Two is for sure.
side camera
Oh, is it? Okay.
there?
Cool.
middle.
Yeah, so because we're on a slope, and the way this heat flow probe is mounted onto the side of the sub, it's going in vertically, but whoa, what is that? Cool. Okay, I don't know what that was. Isopod?
Yeah.
Good guess. All right. Who is that, Kyle? Thank you, Kyle. Okay, Kyle's a real biologist. Okay, that was an isopod with its little flippers, it looks like it's wearing. So what we get while this measurement is happening, as not exciting as it is for you, we're actually getting live data from the seafloor that's showing up on our laptop in the control room, and we can see how that temperature measurement is changing over time after the frictional heating. And then we'll also be able to see that heat pulse and get a sense of what kind of what range that value is going to be, but also make sure that that instrument is working and functioning properly. Because we are on a slope here, so the way it's mounted onto the side of the sub, of the 60 centimeters, we're probably missing the top sensor, but that's fine because we have 8. And to get a really good sense of how temperature is changing, we probably really only need 3 or 4 of those sensors. So
Always good.
Oh, good. Okay. How was lunch?
Skip lunch.
Don't want to waste away. You don't want, you can't skip lunch. It's too important. So you don't know how good it was. You don't want to know. Okay. Oh, let's see. What's that guy doing over there? That is a halosaur, I think. Just keeping an eye on us. Not really sure what we're doing. Yes. Yes, I'm not joking. Like 3 years ago, I couldn't tell a rock from a sponge, so
That one's definitely not a plant.
that is that is not a plant when it's moving, but it could be, Rob. I mean, yeah, Rob and I are the 2 people on the ship of the science party who are not biologists. So, hydrogeologists and geophysicists. So we stare at data, but we prefer to stare at animals like everyone else. Is it a scrolly thing? Okay. What are those like?
It's like
What's it?
like some modifications on spines to
Yeah.
protect themselves.
So what do those do to protect them? Like what are these little? The spines are for protection, but what are these little?
Yeah, there's like other kind of spines. It's
Oh.
modified.
Okay. So the researchers are on this ship. We have 3 sponge people, which is very cool. I don't think I had as much appreciation for sponges until talking to them. One of the groups is working or has historically worked closely with medical researchers, because a lot of the medical advances that we have undertaken are the result of sponges and materials sampled from sponges. And a lot of these creatures, because they have to adapt to really harsh living conditions, really low energy, they have to defend themselves. And, you know, for sponges, they've been around for 500 million years on this planet. So they've had a long time to adapt to different conditions and develop tools that are really useful in medical science. And as a result, there are cures that have been developed from their tissue, which is really cool. And that's just the sponges we know about. We are, you know, I think that the estimates are, you know, what we know 10% of what's alive in this at the seafloor. So certainly there are new medical advances to be discovered from critters that still haven't been seen or sampled. And I said this once before on a previous dive, but as someone who's more of a physics person, sponges are, glass sponges especially, are wild in the way that they develop and form their spicules in this lattice pattern that is apparently studied by material scientists and architects for the mechanical properties of what got pulled. How's the probe? Looks good. Did you see anything from that?
Yeah,
No?
It's the cooling curve at the heat pulse is
Little wobbly. Okay.
So we can
Yeah.
probably end that measurement.
Okay.
We can't model it.
Okay. But that just means the thermal conductivity aspect,
We'll use
not
the
the.
last one.
Yep. Okay. We're good. All right. So we're ready to start the transect and pull up the probe. Not in that order. Sorry. Probe first. Cool. All right, so who's up on this thing? Oh, Kyle. All right. So Kyle is a brilliant student working with Dr. Diva Amon, who just had his first publication come out yesterday, and we are all very excited about it, as should you be. And I'm going to hand it off to his capable hands.
So we're just heading to waypoint 2, correct?
I think so. Waypoint 2? Is that where we're?
Yeah, we're heading from waypoint 1 to waypoint 2.
Cool. Awesome. Yes.
You okay?
Yeah, right here. Okay, right. So we will be making our way from waypoint 1 to waypoint 2.
Yep.
We're gonna do a 300-meter transect
From waypoint 2?
from waypoint 1 to waypoint 2. So like,
From
well,
here, or you want me to go back to waypoint 1?
if we could possibly go back to waypoint 1 to just, yeah. Is that okay? If that's too much.
We're only 100 meters away from the start of waypoint 1.
Okay.
So you want us to do the transect from here?
Let's do for now. But
Kyle, yeah, when you get a chance, we can turn the power off.
Oh, yeah.
Yep, okay.
okay, right. So we don't have to go back to the start then. So we can go from here. A 300-meter transect, and then I'm sure we finish the 300 meters before we reach waypoint 2. But
Yes.
yeah, we'll put markers every 100 meters of the 300-meter transect. We'll flash the lights at the beginning and flash the lights every 100 meters to signify the every 100 meters. And then it'll be like 1 to 2 meters above the sea floor, and yeah. Thank you. Can we turn off the power on the probe? Thank you. I know it's a lot, but just okay.
Sea flow off.
Well,
It's just setting it up.
okay.
Great job, Peter. Good boy.
Okay, great boy.
And can we have the lasers on as well? Thank you. I'll let you know once it's over.
Okay. Oh, the diving. I mean, yes, and everything's
Transect started.
Anyway.
Alright, transit started.
Great. Great, okay. So,
Okay, good. Well, it's afternoon here, but hello to everyone who's watching, and greetings wherever you are in the world right now. My name is Kyle Foster, and I am a student at the University of the West Indies St. Augustine campus in Trinidad and Tobago. And today we are exploring Trinidad and Tobago's deep sea. Currently, we are exploring some known methane seep environments that have been mapped about over a decade ago, back in 2014. But today we are visually exploring these areas. So right now we're conducting a 300-meter transect between waypoint 1 and waypoint 2 as we make our way up the slope of this mound. And we're conducting the transect to basically understand species distribution within this area and to map the change as we go along. So changes in species composition, population dynamics. We're just getting an understanding of what exists within the area. And we're also keeping our eyes peeled for bubbles. So bubbles of methane coming up from the seafloor. And down in the deep, sunlight does not reach these depths. So you might be wondering how exactly is life surviving in these types of environments. So instead of, like how we have on land, plants using sunlight to photosynthesize and make that into energy, we have organisms down here that are able to use the chemicals that are being emitted from the seafloor to be able to transform that into energy. So that creates very interesting communities around these seeps. So we'll be conducting transects throughout the area. We'll also be conducting some biological sampling. We want to be able to understand how, for instance, microplastics or heavy metals are impacting these organisms that live down here. We also are making a point to collect species to be able to display and house locally at the UE Zoological Museum, so that Trinidadians can see and connect with the type of deep sea biodiversity that exists within their own backyard. So currently we're at the base of this mound, which is quite sedimented. It's clay-like or muddy sediments. And well, I anticipate as we move towards the peak of this mound, we'll find more rocks and harder substrates. And that's probably a change in the biodiversity as well. Also, as we do this transect, the pilots are fighting a strong current, so we're very grateful for them and the skills that they have to be able to work through this. A nice sea star there on the right. We're also seeing, bits of sargassum scattered around. And for those of you who live in the Caribbean, you're quite familiar with sargassum seaweed and how it's been inundating our beaches. Well, since 2011, really. So it's quite interesting to see how it interacts with the deep sea and the species that interact with the sargassum. Now, as we're doing this transect, you'll see two green dots. Those are laser projections that are spaced 10 centimeters apart. So it gives us an understanding of the size of the organisms that we view while this transect is being conducted. And thank you for the congratulations, really appreciate it. And we're really excited to be able to do, such research. This expedition is really important for Trinidad and Tobago. My colleague Jamie Lee Luchin would have recently published a paper that would have assessed the state of knowledge of Trinidad's deep sea biodiversity. So from her research, she would have revealed that about 451 species were known for Trinidad and Tobago's deep waters, and estimations showed that about 70% were still left to be discovered. So that's quite a lot. Passing a glass sponge and a sea cucumber there. So yeah, that's why this sort of work is important, because there are a whole host of activities that are taking place in and around Trinidad's waters, and sustainable management decisions that need to be made. So if we are to make the right decisions, we need to understand what is living within these waters, to be able to accurately assess and make decisions. So we're currently about 1,290 meters below the surface of the ocean. And we'll get higher as we make our way up this mound. And we're keeping our eyes open for any bubbling from the seafloor.
Great, Jarvis. Yeah, so I don't think they've got better, so probably we could come back up south. You could come maybe launch point alongside us. Okay, Roger. I'll update you guys later.
Sorry, I know you're back on your lot right now. Could we also flash the lights while we reach each marker? Thank you, sorry. Okay, so I'm sure you can all tell that there's quite a strong current. So the pilots are doing their best as we move along. We would have had a quite exciting few days that would have just passed us. Yesterday we would have explored an ancient coal seep where we saw a species of octopus, or genus of octopus, known as the Granulodoni. So it's possibly a new species which we have collected, and we'll spend some time. A taxonomist will be describing the species so that it can be properly classified. And yes, I agree, the pilots are doing great. Got a shrimp there. Two different species of shrimp. But yeah, so we're doing these transects so we can get an understanding of the changes in communities as we move from the base of this slope towards the top. That's a hollow sword there at the bottom. Largely soft sediments within this area. Looks like a type of coral coming out. And a glass sponge off to the left. But as we go along, we're seeing more and more rocks. And these rocks are host to a variety of sponges and corals that need these hard substrates to be able to colonize.
updated waypoint bridge.
Okay, wait.
Roger.
And if you're just tuning in, the two green lasers that you're seeing, the two dots, are spaced 10 centimeters apart. And these are important as we conduct our transects because they help to give us a sense of the size of what we're seeing as we move along. So this area has been explored previously, twice in the 1980s and 2014. So these are some methane seeps that were explored before. While here, we would have spent the first maybe hour or so conducting heat flow measurements. And we're making our way to sound waves, and those sound waves bounce back and they paint a picture of what the seafloor looks like. So the same way how we have mountains and valleys on land, the seafloor is also very varied in terms of the features and the structures that it has. You know, it's not just one flat plain area. So using the multi-beam, we're able to understand the different structures that are found on the seafloor, and we can make a decision as to whether we want to explore the area or not, because, you know, with different features, you'd have different types of biodiversity that are associated with these areas. So seepage was recorded using multi-beam in the past, but it's not been visually observed. So that's what we're doing today. We're going to see for ourselves and confirm whether or not we will see some bubbles. And so already within this transect, we can see the change of the seafloor from just muddy sediments to, you can see some rocks now. We're coming on to the end of the transect, but we're seeing a lot more rocks than we did at the beginning. There's a nice little ridge there, and we're making our way up. And then once we're at the end of the transect, we'll just flash the lights, and then that will be the end of it. And then from there until waypoint 2, we'll be keeping a lookout for any bubbles, any... Yep. No, not yet. I'm wondering if those are clamshells that we can see. Some dead clamshells there. Those white bits. But we'll continue the transect and conduct some exploration afterwards. And we're now above, or just above 1,270 meters below the surface of the ocean. Let's say just below 1,270. Okay, so now 100 meters after this.
Okay. 1, 2, 3.
Okay.
You guys take the data from this, like from the map?
Not sure actually, but hopefully, I think it'll be good to have.
Do you want to try firing this? Going
I
nowhere.
was about that.
That could be stress.
Bridge. We just slow down a little. Let's catch up.
Slow it down.
All right, so we've got another 100 meters of the transect. And if you were with us from the beginning of the transect, you would have seen that we started off, I don't want to say plain, but you know, just muddy sediment, and now we're moving into a more rocky environment. And that's the importance of these transects, being able to document the changes we go along and the changes in the biodiversity that we see being associated with these environments. Nice black coral there, spiraling on the rock. And then dotted all along the rocks as well, those like little white spots are sponges. So see sponges encrusting these rocks. Some more corals. And then once again, for anyone tuning in, if you want to understand how big these organisms are, just have a look at the two green dots that you can see on your screen. And between them would be 10 centimeters. So we're now up over another ridge. Say being close to the seafloor is too risky, given the currents and everything.
It's not the seafloor. Using umbilical is more pulling us away from the seafloor.
Mm-hmm. Oh, it's supposed to be.
Footage is okay still.
It should be, but the elevation obviously we're hoping could be like 1 to 2 meters above sea level. Yeah. But again, with the
flying
right
surface, which effective studies
to do transects in this condition. Understood.
It would be much easier for us to like stop and jump ahead, look at something, stop, and just flying. It's more harder. But yeah, probably that could
Mm-hmm.
I'm not sure if you guys got any quick footage. If it's good enough. I mean, it looks good enough.
Well, there's something over there for our meeting this evening. And would you say that's particularly because of the conditions? Yeah. Okay, well, we'll just try to finish over this one and then. And then we'll just flash the lights to signify the end of the transfer. Thank you. Just finish the transact. Yeah. Thank you very much.
Yeah.
Okay, everyone, that's it for me. I just want to say a massive thank you to the pilots for conducting the transact in these conditions. And I now hand over to my colleague Giuliano.
Okay.
Hi everyone, my name is Juliano Palacios. I'm an independent researcher working on climate change effects on the oceans, and I'll be with you for the next hour or so. Hola, hola a todos los que nos acompañan de países hispanohablantes. Mi nombre es Juliano Palacios. Yo trabajo principalmente con cambio climático en los océanos y los estaré acompañando en la próxima hora. So you only have a sampling hit. Hitless. And it's literally called sampling hitless. So
You guys want this now?
basically, yeah. So
After
basically,
we saw so many yesterday.
yeah. So yesterday we were not collecting, and so today the main objective is.
Let's hope to see them.
We hope to see them. And the idea is to get also some like 10-second videos in each one of them, like very zoomed in videos. Zoom in once we got, then collect.
Like the samples.
Yeah, all
Pick
the
them up, video.
Yeah, it doesn't necessarily have to be picked up, either in there, yeah. But just with the lasers and without the lasers. We'll see what we get. So for those who just join us in the middle of the dive, we are exploring a region of methane seeps that we're actually exploring in the 80s and in the early 2000s. We're coming back to the site. And we are close to our second waypoint upwards in the dome. And for those who were with us yesterday dive was also in this same region, not exactly in the same spot, but in the same region. And we mostly did visual exploration with no collection, whereas today we will be focusing on collecting some specimens. Many of them are expected to be new to science. And so the reason why we need these specimens is to properly identify them. Para aquellos que nos acompañaron ayer, el museo de ayer fue principalmente de observación, mientras que en el de hoy vamos a enfocarnos más en colecta de especímenes. Muchos de los especímenes que pretendemos colectar creemos que son nuevos para la ciencia y entonces es necesario colectar al menos 2 o 3 individuos para hacer una identificación correcta de estos y posiblemente descubrir nuevas especies. That's the current.
Which are the waypoints that place each of these. Which are they?
Sorry, I thought I pressed it button. You wait, button.
An octocoral. Nope, a brittle star, and we pass it moving along the seabed. If you're wondering why it's so moving, is that we have really strong currents at the bottom, and so that means Sebastian and more movement, that wavy movement. A large black coral. And yes, actually today up here on the surface is also very wavy. The Falcor is, it's really moving today. Some of us are feeling it. We got a couple of fish. Yes. On the top there of the screen. So that is swimming across the screen, but I cannot see exactly who it is or what it is. Black corals, you can see this spiral-like organism.
not witness. Picture of it, good observation.
Wait a second.
Pull up on the page.
Is it correct?
Hmm?
Is it the correct or like stick?
Yeah.
Oh, can we see that crab, please? That's one of our
Catch.
yeah catch of the day. That is a Shakyun Quinquidens or Finary. So we can get a very good look at it. Huh?
Are you gonna try to get it back?
Yeah, well, I mean, it's.
I know, are you gonna try to get it?
Yeah, we're gonna try to get it, yeah.
Net.
Yeah, probably with the net and stuff. It's the easiest. We can try, but we don't need to like chase him down until the end of the the dive, because there will be
Well,
plenty more. Yeah, that was the. Yeah, there will be lots of crabs in the sea.
whatever you guys have. Are you gonna put it straight? Like, just say so the handle is up. Huh? Goes through?
There's holes in the bottom of the basket.
I'm gonna try to grab it.
Oh, watch it out too, so I get the that.
Okay.
Why are you grabbing that?
Something nice.
You could write orange handle. You could take the camera however you like. Because I'm using this camera to watch you grab.
live in 76 of them.
Hmm?
So we're gonna need 6 of those.
Yeah.
So, you see how long it takes to do one?
Yeah.
So I think we may get it, but eventually go up on the seat itself. It's gonna be about an hour.
Yeah, so I
And
heard that.
so we can sort of grab a bunch. Here's here, this is the line. And then it goes in the box. And then jump on.
Okay.
So-
Well, but I think I'm getting in the net.
Actually,
Yeah,
Just
that's
stay in the
correct.
net.
And then, you
So,
know what,
so
and if-
try this one, if not, we'll continue.
Yeah, exactly.
Yeah. The other ones. Yeah. Yeah. Right, so we're gonna try to sample the scrub with our net. And then we'll continue our dive. Yeah, it's all good.
Don't go down too much. I'm coming down. Grab it. Grab it.
Grab it.
Grab it. Grab it. Grab it. Put up your arm a little. Put up your arm. It's in it. That is good, right? Just leave your arm just like that. Clockwise. Come on, man. Come on, man. You got it. You have it.
You got it.
Come on. Come on. Right, two.
Oh.
Alright, you set your arm. Fix it. Come up a little. What?
So there will be other crabs in the sips, and technically they will be more abundant. So we might
Want to give up on this one or one more try?
let's do one more try and then.
Alright, no time to shine.
So,
You have a, you
No
have
pressure.
a net full of rocks right now. You want the crab, not rocks. Oh my. They should find the cameras because so I can fly a little better. Yeah, I will try it one more. Yeah. Okay. And yeah, this dark metal mud.
I
right
like
in there.
Philippine.
Come in center, honey. Center the screen. All yours. Come on, come on.
It's getting in there.
Chop a little, chop a little. Alright. Abort.
All right, let's continue to the seep. We'll find more.
Just leave them alone.
Yeah, we'll find more.
At least we got a net full of rocks.
All right, we will continue our way to the seep.
Put it on the side. That side. Great, you could continue that move.
Animate.
You could take out that. You could empty it. You don't think you can.
Sea cucumber on the lower left side of the screen.
That's something. Come on, just like 1 minute. Come on, like 1 minute.
Let's continue. From the seeps, they will happen more abundance.
I think I got it all there.
See a fish on the top left of the screen. And we'll keep going to our second point.
flying nice.
It is flying.
Because the vessel just started moving. The vessel was stopped. To probably pull it in, just put it on the side. But we need to give it a bottle box for that. You could leave it in your arm for you while. Just
I'm gonna like, yeah, take it away
take
from
it
the
away from
main
the
camera.
Somebody's asked.
To take the net away from the camera, yeah. Thank you.
Put some time.
Can you like store it?
Big one. On the bus one, sorry.
Yeah, just because so it won't be in the screen. Thanks. The purple one? Purple. All right, thank you. So we can continue now.
average waypoints update.
Update waypoint.
There's a fish on the left of the screen. More black corals. Seems to be sea stars. Small sea stars.
Good luck.
Yeah, bouncing is another statement. Thanks, Tom. Yeah, I believe. É, a gente tá indo pro waypoint 2. Chegando lá, vai.
Me? So you just sneak in there, huh? Now you done did it. This one better? We're just leaving that light off so you can see who bless. No, so when you're on deck, you gotta, I think you should swap that with another light and then try to plug it up.
Can you zoom into that? Can you stop? Yeah, right in the bottom. Right with the laser bar. I don't know what's that. Yeah. Okay. It's quite cold under the rock. I think we're gonna fly and get it from around. Yeah, we can switch. Wait, it's not what I thought it was. Flash function after waypoint. So it's
Hi everyone, Robert Cassidy here, PhD student at the National Museum of Natural Sciences in Madrid, taking over for Giuliano. Gonna be with you for the next hour while we continue making our way towards the seeps and seeing what we find.
You good?
Yeah. So we've started off a little bit of a ways away from the main seep site and are working our way towards it right now. Seep site we're going to have been explored twice before, once in the 1980s and once in 2014. But as we were saying yesterday, these areas tend to be very diverse and we see new stuff every time, right? For example, we saw what we think is a brand new species of octopus yesterday within a pretty short time of starting our dive. Got some, I think, black coral right here. Yeah, so as people might have said already, these methane seep sites have a very different chemical and geological makeup from most of the other areas that we've been in, are pretty unique types of ecosystems generally in the deep sea. The closer we get to the actual seep site, where we hope to find bubbles coming actually from the seafloor, the more we can expect the faunal community to change, right? Because for most animals, these seeps are toxic areas. They're very anoxic, low in oxygen. There's all kinds of other chemicals in the water, but there are entire ecosystems that are very abundant, very diverse, that have actually adapted specifically to these areas. We found just that yesterday when we got very close to the kind of peak seep site at Dome 1, and we saw just these huge beds of giant mussels, right? And these mussels are things that you often find at seeps and at hydrothermal vents. And they are interesting because they are chemosynthetic. I think many of them rely on these very unique symbioses with bacteria that actually consume the different chemicals that come out of seeps and are adapted to the area. So these types of ecosystems are pretty it's a pretty new field of exploration, right? They didn't know they existed until the 1970s, so it's not surprising that pretty much every time someone goes down to one of these sites, we find something new. So right now we're kind of in the area of bubble streams that have been mapped but not visually confirmed. And when I say bubble streams, what I mean is I'm referring to the bubbles that come up from methane seeps, and it's actually possible to see them on the mapping that we do at night, which helps us, decide on areas to explore. But it's also good to visually confirm them and get a more precise idea of exactly where they are, because these bubble streams are where we, hope to find those very abundant communities, things like mussels. Got might be seeing some sponges here. Could you try to zoom in on that white circular part on that rock, if you can? Yeah, the current's kind of strong today, isn't it?
Yes, please.
Okay, yeah, that looks like a sponge. I think you want to.
I think at this point I want to just find
Yeah.
a seat.
Okay. So we can just keep going. But maybe we just get a little bit more footage. Oh, possible.
Yeah, it's just, yeah. And then more footage, it's just, if you want. I was just looking at it.
Okay. Did you get some frame grabs, Shauna? Few. We could try like maybe one more zoom, if possible, and just get a quick picture, and we keep going. Yeah, that's good. Yep. Just spam it if you can. Okay. Yeah, I think we're good. Thanks. Yeah, so sponges are definitely one of the things we're interested in at this site, as we are with most sites. But right now, our priority is trying to locate the actual site of the seep. So we're going to keep going. The hope of getting to a site that we believe is there, but has not been visually confirmed before. So when we're getting samples from these places, you know, every sample that we get from a place like this is extremely valuable from a scientific perspective. Not only are we kind of counting, categorizing the different animals that we find at these sites to get a general picture of the biodiversity, but each physical sample we get is also extremely valuable for genetic work, for microbial work, for museum specimens. A lot of these specimens are going to the Zoological Museum at the University of the West Indies, where one of our expedition mates, Zahir, is a curator. And all of these different components kind of form part of the more modern idea of taxonomy that we have, which is what we call integrative taxonomy, and you know, relies on an understanding of not only the visual morphological characteristics of a species, but also, you know, its distribution and specifically its genetic makeup as well. So when you get these marine invertebrates, especially things like sponges, maybe echinoderms also, a lot of them can be extremely plastic in how they look. One sponge might be one species but have 10 different colors in 10 different places. And so it's important to understand you know, the different ways of distinguishing between species for things like these. They kind of test our idea of what is a species a lot of the time. We've got Tina 4 floating by. And it looks like some kind of big crab or squat lobster in the distance also. Got another big crab over here. We can see some tube castings of some polycute worms. These tube worms are one of the most common things you see at chemosynthetic sites like this. Oh. What are you guys measuring?
No, I think just it fell from the
holder.
big crab walking by.
On the camera, right?
Beautiful crab here. Yeah. And we're getting more of these large kind of examples of these worm castings we were just talking about. This to me indicates we're getting maybe a little bit closer to the seep. Maybe if we follow the crab, it'll tell us where to go.
If you don't need it, wrap that pan up. If you're sitting, you can see like what's way out too. That's the side.
Another small crab and a shrimp, it looks like, hanging out. You can see there's a very steep slope as we're kind of working our way up this this dome. Looks like another large tube worm here.
So would it make sense if these mounds are caused by the seeps, that the highest point would be a likely place for them?
That's what I would think, yeah. I guess that's where we're kind of going, isn't it?
Yeah.
Yeah.
So
I mean, I'm not a geologist, so I'm not really sure, but it seems like that would be the case for sure. Yeah. So these seeps are also called cold seeps, and that's because they're kind of like the counterpart to hydrothermal vents, right? So hydrothermal vents can, be some of the hottest places in the sea on Earth sometimes. Very hot kind of plumes of chemicals and kind of smoke coming from the sea floor, whereas cold seeps like this one don't actually have a real difference in temperature, but they do have these excretions coming up from the floor. Oh yeah. Okay.
It's promising.
Yeah.
Stuff.
Probably going that direction, right? Yeah. So you can see, yeah, kind of going up the slope to the left. Well, I guess that's where the route is, right? We're just riding the current right now. It's a pretty strong current, kind of stormy-ish seas today. Wow, look at this huge rock just full of these tube worm casings. That's very promising for it's an indicator that we're getting closer to the seep site. These are massive. Yeah, these tube worms, they form a lot of structure in habitats like these. They're very important for the ecosystem. Another large crab here.
If you turn to port, just kind of work down that ridge and just see if there's anything crazy.
Yep, just having a look around while we continue seep hunting, seeing what clues we can find.
Super odd. So there's a big white something over there. But
Yeah.
Should we check that out? What do you think?
Oh, yeah, let's top the hill, right?
yeah, it's off the hill.
Yeah, let's just do that.
You want to go towards that?
Sure.
Okay.
Check it out.
See what she's talking about? Just behind the boulder.
Yes.
Right here, backs. Touching something. I'll move the boat down toward you. Bridge, RV, waypoint.
I'll tell you, waypoint.
The big casing boulder. White patches on the seabed, other than that. Good sign.
Oh yeah.
Just as you're going through, you're probably already paying attention, but just make sure that thing doesn't get hit.
Oh yeah. This rod thing here?
That's right.
Yeah. I'm sure you guys know, but if we just try to keep that probe safe from any bumps. I know it's tricky. Yeah. I mean, it looks like we're doing good right now. Seeing, I think, various sponges on this rock
Yeah, sure.
alongside all the tube worm casings.
Oh, there's a few large ones, huh?
Some kind of
Oh, there's a bunch of large ones.
Yeah.
microbial mat as well.
Sponges.
Yep.
The darker things over there.
The darker thing is probably sargassum trapped. Like it will issue.
Yeah, wow.
We're seeing bubbles.
So with sponges like these, we can expect to have a really unique microbiome that is specifically adapted to a seep environment. I think there's actually been papers on studying different species of sponges and of other invertebrates that have, despite being very distantly related, their microbiomes have shifted towards the same composition at seep sites, just because having these bacteria that can process these nutrients is very important. Yeah. And can we zoom in just a little bit more on that kind of ground area? We're gonna try to look for bubbles here if we can. Yep. So my friend Robert, my college roommate, is asking what's the delay like on the controls, and it's actually pretty instantaneous. And you can tell that it takes a lot of skill. We've got some very talented ROV pilots here, but I think it's a pretty much instantaneous reaction from the ROV. We've got fiber optic cables that go all the way down. And we're looking here, trying to see if we see any bubbles. I'm not seeing any so far. Wow, very interesting though. You can see like just the tip of this worm poking out of the casting.
Purple fuzz.
Let's call this up. Yeah, the purple fuzz is
We're trying to figure out what the purple fuzz is too.
have them at vents as well. It's
So we've got some very weird kind of microbial mats here.
yeah.
Maybe even some sponge tissue as well.
You
In
mean
some of
the
these
purple?
nooks and crannies.
Oh, it's says with an F.
Maybe it was the right. That's what gas we call it. Call it G.
Yeah.
Those kind of feathery, they look like feather dusters poking out. Those are actually the worms as well. They've got these kind of fan-like mouth parts that help them grab things that are floating by. Beautiful.
Okay,
Yeah, thanks.
carry on.
Yep, sounds good.
Okay. I'm just gonna go towards 30.
Now we're back up, continuing our hunt for bubbles. We've diverged just a little bit from our path to waypoint 3, just trying to explore these different areas that have potential to lead us to the seep site.
No samples yet, huh?
So this area that we're in is one of the maybe the only one of the few explored areas that we're stopping in on this entire expedition. And that's because there just is so much interest in better understanding what this habitat is like, and what else we might find here, and identifying exactly where these seeps are coming from. This is important because like these areas host very unique biodiversity, but they're also of pretty big interest for industrial applications like oil and gas. And it's important to know exactly what's down here before, you know, it is potentially used in any kind of extractive sense. So this will help us manage conservation in the future, and hopefully prevent any kind of overly destructive activity in areas like this. If you think of the, you know, some people ask why it matters to explore these deep sea areas and why it matters to try to conserve biodiversity in these places. And I mean, there's a lot of answers for that. One of them is that we're talking about habitats where not only are these animals adapted to just the inherent massive hydrostatic pressure of being at, you know, sub 1,000 meters, where any of us would just explode if we were down here. But they also have really unique adaptations and microbiomes to places like this that are extremely valuable for anything related to biomedicine and biotechnology. If you think of, you know, the PCR tests that we all did during the pandemic, that molecular process that allows us to identify things on a genetic basis, that would be impossible if it were not for some biomolecules discovered at a hydrothermal vent in, I think, the 80s. And that's the case for a lot of different types of, you know, very important applications. Sea sponges, for example, some of the first drugs to treat leukemia and HIV were derived from sea sponge microbiomes. So if you're, you know, asking if you should be interested in conservation in these areas, it's really a question of, you know, you don't know what you have to lose unless you explore it first, right? So we've got some pretty big rocks over here. Big one on the right. Oh, I'm seeing more stuff in the distance here. Could be promising maybe some more of these stylasterid corals. Got a question on whether we are sampling these sponges in this area. And so far today, we've not really sampled biologically. Our main goal right now is to find the seep site and hopefully sample there. Yeah.
Go a little bit over, then I don't.
Okay. Yeah. To hit the waypoint.
Just to hit the line.
Yeah.
Rather than go like this.
Okay. Anna was asking if we could maybe, instead of cutting the corner, try to go up to the 3 marker and then get the line from there. Do you think that's possible? Or is it kind of tricky? It's great. Beautiful coral here.
We can pick that up, big one.
You want to get a piece of this?
Sure. Might as well get some.
Okay. Yeah, if we could get a piece of this, that'd be great. Doesn't need to be the whole thing, just like 2 10-inch pieces. Okay. Okay, so we want to try to get, if we can, 2 branches about that long, and they could be in the same quiver. So we just coral quiver 1, I think.
Coral quiver 1?
Yep. So we are going to try to get a sample of this coral. Since it's a very large specimen, and we're here anyway. Okay. Cool. And if we could just try to center it for a few seconds before we sample, just to get some photos. That'd be great. Okay. Yep. So if we could be here for a few more seconds with the lasers on. Or we get some good photos. Spamming. Yeah. And yeah, I've got a cool brittle star, maybe a few there. Some crinoids as well. Now if we could do the lasers off for a bit too. Great. Wow, beautiful. This is a huge coral. So those lasers that were on just a second ago, those were 10 centimeters in width. So that gives you an idea that this is a very big coral. You can even see all these little polyps, right, all along the branches. Is this a hydrozoan? Not sure.
A crinoid, you mean?
The
big thing up above?
Yeah. Well, do you know what kind of coral it is?
Well, some crinoid.
Yeah.
Some crinoid.
Yeah.
This is a crinoid.
Yeah. So that's a crinoid. Crinoids are actually echinoderms, right? Yes. Yeah. So they look like a coral kind of, but they're very closely related to sea cucumbers, sea stars, urchins, and brittle stars.
Like that one down there.
Yeah. So that's great. I think we're good on images now. So whenever you guys want to try to sample, be my guest. So my friend Robert's really liking this coral. Just a quick congratulations to Robert. He got married a few days ago. This is a science stream, but I think it's pretty important too. This isn't a Gorgonian, right, Anna? Anna, this isn't a Gorgonian, right? It's not a Gorgonian. It's coral.
You want two pieces, right?
Octocoral. Some kind.
Two
Yeah.
pieces in the
I mean,
it is a Gorgonian, but we don't use that term.
Yeah, so 2 pieces, if they could be like about that long, each one, that'd be great. Wherever is easiest for you guys to take from. Nice. Yes, please. Just one quick pause. Close up. It's great. If we get some snapshots. Wow. Yeah. I think it's okay with lasers off. You want them on? The longer one? The next one? Okay. Wow, beautiful. I think that's probably good. Thank you. Do you have the number, Shauna, for that sample?
Yeah. 139.
Thanks. Yeah, well done.
That's the bonus piece.
Yeah, so we get one more, a little bit longer if you can than the last one, if it's possible. Yeah, that could be good. Like maybe that one.
Yeah, I'm just trying to think of a long one that would still fit in there.
Right.
Maybe this guy.
Yeah,
That's
we were
the
thinking of
one.
that one. Yep,
That's the one.
perfect. Oh. Got our shrimp floating by. Just curious. We do, yeah, a little bit less than that. That's good, I think. Yeah, I think that's good. Yeah. Thank you. Wow. Beautiful piece of coral. You getting photos, Shauna? You getting photos of this? Thanks. Just checking. I think that's good. Yep. Thanks. So these samples, we just try to take a piece that's big enough to get all of the morphological characteristics that we need to identify the species. While also not taking so much that the individual cannot, you know, regenerate afterwards.
See all the particles flying by?
Yeah.
Bit windy.
Half an inch too much.
Oof. That's okay.
You're too close to the sun.
Okay.
But they have to be live with it all night.
It has to be live, you said?
Yes.
Yeah. I mean, that one looks live over there.
No, I mean a clump.
Oh, a clump.
That's just you know, we're looking at
Yeah. With like the feather. Yeah.
These guys.
Okay.
Comes up right against the rocks.
Do you want to try to do that now or just when we see it?
Let's take a look while we're sitting. If there's anything around.
Okay. Cool. Yeah, so we just want to get like a look around while we're here and see if we see any of those tube worms that might be alive. So if we do find some, we might try to grab a piece of that too. Yeah, these all look like they're not alive right now. Yeah, I don't either. Yeah, I think we're gonna keep going. Okay, so we've got what we need from this little stop. We're gonna keep going. Continue bubble hunting. Got another coral down here.
Okay, wait,
Wow.
but
Clams. Is there clams?
You mean in the distance? Looks like a sponge here in the rock. Maybe some clam shells in the background.
Those are shells, aren't
I think so, yeah.
they? I've got them there.
So if we could go up there, maybe. Big crab as well, hanging out.
Just there we have
These look like rocks to me. I don't see the tops coming out. Yeah.
Clam shells.
Yeah.
If that's a good sign, that's what we're seeing this thing.
Yeah. Yeah, so we're seeing some kind of scattered old clam shells over here. Big crab on the left. These shells are a good sign. We saw these yesterday when we got closer to the seep as well. So kind of starts off with clam shells, mussel shells, and then live ones. Got huge beds of mussels yesterday. Do you want to keep going up here or go back on the path? Anna? We're kind of veering off, yeah. What do you think?
We're gonna explore
Okay.
to the west.
Yes.
Yeah, that's great. Yeah, just keep looking in this area. Yeah. Keep going like up the slope. That'd be good. See some more clam shells scattered around. That's good. I mean, I'm guessing they've probably like shifted down from further up, right? So we want to go up the hill. Yeah.
We can try coming back.
Gotcha.
Let's see what it's like first.
Cool.
Going this way?
What do you think? Back on course? Yeah, I think I want to go back on course now.
Back on the line?
Yep. Thanks. We do like a diagonal towards the line so that we can see some new stuff on the way.
Yeah. Just kind of go like this.
Yeah, a little hypotenuse type thing. Wow, beautiful shiny fish there. And something else. Wow.
Is that under that rock?
Yeah.
Yeah.
Seeing microbial mats here.
Alright. Keep going.
Yeah. Got a big coral here.
A really big coral in the background there.
Yeah. Wow. Big coral. Corals. Wow. Yeah, look at that. That is a massive, wow, multiple massive corals. Whew. Can we get the lasers on just to get some scale?
Yeah, let's take some images.
And some photos too, please.
Laser on.
Wow, okay.
Is that the same kind we just sampled?
Yeah.
Looks like that.
Yeah, it looks like what we just sampled. I mean, that's a 10 centimeter laser. That is a big, big coral. Wow. Covered with brittle stars too. Wow, yeah. Huge. Even more in the background too. Wow. That is gorgeous. Beautiful.
Just go straight like,
Okay.
coming in the circle again.
All right. On that note, I'm gonna pass you all off back to Giuliano. You can continue bubble hunting with him. All right. Thanks. Thanks, guys.
Like you did all the hard work.
Now you get to reap the rewards.
You collected
Sorry,
something.
Jar, we knew what.
You just collected one.
Just one.
And then, which is this one?
Yeah, it's the same kind, but it's more like that.
Rotate. This is yours? Or what's here? Wait. Hi everyone. I'm back. Oh. Yeah. It's a big crab. Is this a spider crab? Do we? Really?
Yeah, it's a paralamis.
Wanna try now?
Not next
Do you wanna try now? Oh, there's a fish in the bottom too.
No, not now.
We can take a good look at it. Estamos viendo o caranguejo grandão que a gente pode passar. Mas pode continuar.
Friend.
Estamos viendo un cangrejo gigante. Y las otras grandes estructuras que son, ya, son corales. Y recuerden que los dos puntos verdes que están viendo tienen 10 centímetros de distancia entre ellos. Eso les da una idea del tamaño de estos corales. Just remembering everyone that the two laser beams that you're seeing on the screen, the two green dots, they have 10 centimeters between them at any single point. So that gives you an idea of the size of these beautiful corals. And the organisms around the corals are brittle stars. Esos organismos que ven alrededor de los corales son ofiuros. Muchísimos. Beautiful. Would you consider this a coral garden? Is this density enough for a coral garden? Not yet. Okay. Not yet considered coral garden. But getting there. Our search for bubbles continue, or quest. Beautiful.
Interesting too, it's like one species, huh?
Yeah. And there's some real even bigger on the back.
The little ones, the same species, or recruits. That's what all
All the little ones are recruits. So a gente
Well,
pode
here,
tentar
there's
fazer
some
zoom
because
nesses
they
pequenos.
look like the same
Esse daí ou naquele de lá também pode ser.
species.
Yeah. So we're just zooming in on the small ones that look to be as recruits of the same species, so they're juveniles. Yeah, they just go from the skeleton. Have a shrimp too on the screen. Sí, todos esos pequeños que ven ahí son organismos más jóvenes de la misma especie que están empezando a crecer. Mucho más jóvenes. Los grandes deben de tener más de 100 años. Estas son animales que crecen muy lentamente. What is that in the, can we see like at the bottom?
You sure about it? Two rooms.
Two burns?
Two rooms, yeah.
Big one.
Thanks.
Quick waypoint, but I'm pretty sure the only active seep we know at this place for sure is a 4. So we just need to make sure we're like, exactly.
Just watch out with this thing.
Oh, thank you.
Not to keep the rock. I don't think it's. I don't know if it is the. I think it's just the vibration of the sub. So two worms in the bottom. Those I see them kind of yellowy greeny organisms on the bottom left. They're dead. I've just been told that they are dead. Oh, crabs. Oh, two crabs. Two for one.
Oh, there's two.
There's two. That's what I'm saying. So can we? My collect those. How do you feel about this?
What's happening? I'll try anything once. So you're gonna have to put your left bumper out. You're gonna have to put your left bumper.
Yeah.
Ideally, we want to get the crabs.
So what do you?
We'll try a little bit with it.
I think that top one might be loose.
Like I think once.
Pilgrim says.
Yeah.
Just trying to get those crabs as close to the center between them. So if you can, that's it.
It's cornered.
I'll get some screen grabs while we are at it.
Oh, don't go in. Don't go in. No.
I'm trying to go here, the current is blowing me.
Protecting his lady.
Well, carrying her around.
Also, she's like kind of kidnapped.
We're fighting against the current at the moment to try to.
I think it's pretty close to as good as we can get. The only other thing you could try is you could try putting the front of your bumper against the rocks. And that'll keep you from having to thrust as much, but we can give it a go. So if I grab these guys' leg, it's gonna pop off, right? That was all I guess. Leg won't pop off? Okay. And you want him in a bio box or where?
Wherever you can put it, yeah.
Both?
Wait, Diva?
Yeah.
Is that in try to rotate this taking rest both drums so that this is
Let me just give it a go.
it's stable but it's blowing.
Just be ready to open that bio box, right? So when it looks like I'm gonna grab them, I'll probably have you go ahead and force out. There's nothing in any bio boxes, right?
No, there's nothing. They're all empty.
So we'll probably go for the big one. You probably wanna send a different picture now in case.
Okay, no excited. They're all empty.
But one of them we're not using at all because
I don't think we should collect amazing bear crabs. I have said that in the chat.
Wait, Zach.
Anna,
I was just pointing at them.
Don't collect it then.
Yeah, sorry.
we don't collect animals commercially when they're brooding, etc., etc. I don't think we should do the same.
So since we're there, can we use like a really good zoom in? Yeah, thanks. Yeah, some screen grabs. Maybe. All right, perfect. Thank you.
Okay?
Yep. Thank you.
You wanna go here or Tobago line?
I think we're good around here. We can continue.
Which one? Which one?
Which one of it?
Which one? What?
No, nothing. We can continue. Oh, look at the wall on the left. We continue in our quest for bubbles.
Bridge, the waypoint. Okay, wait.
Another crab.
Single.
Single crab. Can we zoom in and try to get this one, please? And get some like good video of it. That would be great.
Wanna try to collect this?
Yes, please.
Alright, so I'll try and grab him by the leg. And if that doesn't go well and he doesn't escape, then we'll try and suction sample onto his top and then carry him over and drop him in.
Okay.
Is that okay?
Sure.
whirl. Alright, and you can go ahead and torch out while I'm doing this if you want. So you're ready. Just give me one moment. Saw me, you didn't like that. It's a way better setup than one.
Yeah. Very nice.
He knows just how far I can reach.
Oh. Wow.
Now he's gonna come
He's
close.
like, I have one of those.
Yeah, he positioned himself right at the like
You're not getting me, that's my trick.
1 centimeter away. Oh, close.
Yeah.
Right away.
It's probably on the porch.
Oh, it's going on there.
Under the rock. Oh yeah, there it is. Move on.
Oh, if he's gonna jump,
It's very bad. Yeah.
that's it. It's jumping.
This guy's kind of deserves to go. You keep following him?
Do you wanna just continue for another one?
Sure.
Yeah,
Yeah. He's got it.
he's gone. Thank you for trying that.
This is a really wonderful opportunities.
Well deserved. Mr. Barrett.
All right, so after the trial, we'll continue in our search for bubbles. And just remembering, folks watching, that a lot of the specimens that we are intending to sample during this dive might be new to science. And so in order to describe them and in order to classify them, we need to have, scientists need to have these specimens in hand. Unfortunately, only video or only imaging is not sufficient. This is the main reason why we go through collecting them. Wow. So can we zoom in? Yeah. Have a good zoom in. Huh? you wanted to try to like stop?
Those are not
I'll continue.
what we wanna, if we say a bunch of rules, if we have the kids or them should grab it.
Okay. All right, we can continue. Thank you, guys.
Oh, yeah.
Brittle stars around the coral. Então, kita and to clap. Antes de você chegar, embaixo não tava assim, cara. Ai, não é difícil. Little fish. You can see now in the image the map. We're following our steps to the fourth point on top of the
Yes.
No, nothing. The map. Vamos tentar. Ai. Oh yeah, there was on the left. Do you want to take the whole rock? Oh no, authentic rock. And I see like
These blocks.
yeah, we want to see those, please. Yeah, the small corals. Authentic rocks will come with them. We're trying to see if these two ones alive a collection. A gente tá vendo, mas é esse mesmo, mas vê se tem algum to collect those. Você acha que consegue pegar esses pequenos aí?
RV bridge.
That is the one.
Come on.
Is that
Hey,
this?
Doug, I'm just checking
I
on the
just
map.
want to.
That waypoint big.
You're going mad.
Okay, I'm just coming to a stop now.
Yeah, no
So
worries.
any of these like small white ones? that it's easy to grab. We need two of them. Huh?
You have the right one.
Again, every time I'm in the room, I am mind blown by the skill of the pilots navigating these rocks and landing on these rocks and collecting the specimens. Yeah, and we need like two of these white ones. And if we can have a good zoom in first, that would be great. Oh.
You want to zoom on the little pronoids?
Yeah, the white ones. We need two of them, Zach, please. And you can first zoom into them. Yeah, perfect. With the lasers. Beautiful. Good, Jimmy? Okay, thank you.
Good there?
Yeah.
Yeah. So we can take two, thank you.
Okay, you want these in where? Quiver?
Yeah.
Quiver 3, how about?
Yeah, 2, 3, or 4 works. Currently only one is occupied. Vamos a hacer ahora la colecta de estos especímenes de coral. We're collecting now these coral specimens. They're very, very delicate. Son muy, muy delicados.
Okay, you can basically dim it a little bit. Snap it down, or ice it down, or something.
They're very, very delicate, so you can see how slow the pilot is to grab them. Oh.
Yeah, they're very brittle.
Jamie, we're gonna. He's number 3. And you wanna take a frame grab once it comes into focus. Got it?
Yeah.
Thank you. Oh, he fell in there.
I was guessing not.
You guys not?
I would guess not.
Oh.
We should get extras just in case it do.
Yeah, there's a lot more
Okay.
down there.
There's yeah. Wait for that, Jamie. So can we try the like pala? How do you say pala in English? Yeah. Let's see this one.
We have a scoop, right?
Zach, if we try from under them, like a scoop,
Yeah, let's use a scoop.
and then we can try from under it and then.
Alright, you go ahead and watch out. All the way.
So as you can see, these corners are very fragile. So what we're gonna try to do now is actually scoop them. So it's not only the mobile species like the crabs that are hard to collect down here. The sessile ones are also have their tricks. Skill and patience, it's highly required.
Unfortunately.
Someone number 5. Okay. Anna, what do you think in cuanto a the scoop?
That looks, looks good.
Yeah, that's good. Yeah. Jamie, can you do a crab, please? Got it? Yeah. Thank you. I think I can go to number 3.
You are reel that all the way, or you can do a lot more?
No, they don't want to go. There we go. I don't know. Looks like it. One more?
We don't know if the other one.
I mean, yeah, that was like multiple scoops, so yeah. I mean, it's hard to get just one because they break. Yeah, we're good. That's good. Yeah, thank you, Zach. Thank you. Ah, se a gente pode lá em cima dessa ponta.
Demorou.
Dá uma visualizada. right? After that successful collection, we'll continue searching for bubbles, and we're gonna continue climbing on the top of the dome of the steep to see and explore. Acho que tá, o mapa não tá mexendo mais. Yeah, lots of shells. Can we try to zoom in there? Are they all pieces of shell? There's a crab in there if you want to try it. Some shells. Yeah, good. We can continue going up in that direction.
That direction?
Yeah, that will take us to kind of like the top of the, yeah. No, no, we just want to explore a bit of the top of the, if possible. Not really in here, is it?
No, I don't think so. It just has circular.
So stay away, but
Yeah. Yeah.
There's active muscles.
And then there's
Active
this.
muscles? Oh, there we go. I mean, those are
So,
tricky to
wait,
grab,
let's
but
see.
we can keep going up.
Okay, keep going.
Yeah, there's some mussels in there. There's a crab, and there's two worms as well. So two worms are like longer.
Stick out.
Yeah. Right.
Circulars are attached to the rock.
And we don't want two worms. It's a very small fish at the bottom. Oh, that's shrimp. Yeah, she wants like 20 of those, so we'll probably find them and wow.
Well, it's covered with bacteria.
Yeah, covered in bacteria.
Let's take some imagery, maybe.
Look at the shrimp. Wow. Yeah, if we can get some like nice close-ups. Thanks. Covered in bacteria. Wow. Jamie, you getting some? Oh, screen grab, fish grab. You got a frame grab of the fish with a.
Yeah, I got an observation. I actually did.
That's cool. Wow. Okay. You got good? Really? Oh, no. Thank you. Thank you. We can. Continue going up. That is amazing. That is basically, es un camarón que está cubierto en bacterias. En realidad debería ser rojo, pero lo estamos viendo blanco, blanco, blanco. Está cubierto en bacterias de las cuales se alimenta. Y vamos a continuar en busca de burbujas. We'll continue our search for bubbles around. Is that an anemone? Yeah. Looks like an anemone over there. I think we can get like a visual of the. Yeah, pode crer, pode crer. Seems to be shells.
Anyways, like,
You guessed it. So this green on my screen here is the, Julian was saying that's supposedly the top here, that's what we're trying to see.
Yeah, that would be great if we can like.
Okay.
We don't need to go directly to it. We can like zigzag around it just to look around.
Sure.
That would be great. Thanks.
I think it's about 100 meters off. Well, it's about 30 meters little guys.
Oh, is that a fish? A shrimp. No, it's a shrimp. This one is not covered in bacteria. Some more shrimps.
Update waypoint.
Oh, look at the size of the rock. Big wall.
Speed of rotation. Keep it right forward. A little bit better beginning of where cigarettes in the pretzels and release.
It's like it also goes really slow.
All right, everyone. I'm switching now. We'll leave you in the good hands of Shirley, and I'll see you again tomorrow or the day after. Enjoy the exploration. So we have 2 falls and the fingers.
Okay.
And that's all of it. And I marked I marked the one that I collected, which is this one.
Yeah.
And I think the other one was this one. I don't know if it was this one.
Yeah, I think.
Yeah, but they didn't mark it.
I think that was.
Yeah. Yeah, but I marked
Okay,
it.
okay, so you got the
Yeah.
stylus, yeah,
Yeah.
I think they got the, I think the Pernod. was
Yeah,
the
I
one.
think so. And so we're just
Yep.
exploring here at the top.
Yep, we're gonna go
Before
around and look
going
at that
down
high
the
point and
yeah.
then go on. Okay, sounds good, thank you, Juliana.
Thanks.
Thanks. So apparently what we want to do now is we're both gonna, yeah, kind of explore around the top. And then when we're done looking around the top, I'm gonna go head towards waypoint 4.
And then we have a number of things that we're targeting that we would like to try and collect. So I'll try and alert you before we're right on top of them. Okay. Hi everyone. My name is Shirley Pomponi, sponge biologist, biotechnologist from Harbor Branch Oceanographic Institute at Florida Atlantic University in the United States. And I'm taking over this dive lead for the next hour. We are about in the middle of a 12-hour dive at a site that we're interested in focusing on because of potential methane seeps, and actually we're seeing a lot of the animals that are associated with these seeps. So we're on the lookout for bubbles. It would be really cool to see some methane seeping up from the bottom. This area was explored in the 1980s and again in 2014. Earlier in the dive, Dr. Rachel Lahr conducted some heat flow measurements, and we've climbed to the top, just about to the top, the highest point of this feature. So we're going to be exploring around this area for the next few minutes. And then I've got a list of some target species that we would like to be able to sample so that we can study these organisms a little bit more. And so far today we've collected a couple, I think a primnoid coral and a stylaster coral, so two of the species that we were interested in collecting for further study. All of these samples are going to be housed at the University of the West Indies here in Trinidad, and available for study by the scientists here and globally. I'm one of 25 scientists on board for this expedition. More than half of the scientists are researchers, resource managers, and students from Trinidad and Tobago. So it's a Trini-led expedition. Our chief scientist is from Trinidad, Dr. Diva Aman. And we've got a multinational, multidisciplinary team participating in this exploration.
New waypoint bridge. Let's bring you west on this.
Shirley?
it's on like a circular thing.
It is, oh, it's on some, yeah, let's take a look at that. What is that? Oh, it might be just me rare. I don't know.
Drop weight from something, or like,
Yeah, looks like it's,
boring.
looks like something that was,
Ooh,
what's on top of this chain?
somebody's weight.
Looks like maybe chain or rope lines.
yeah, it looks like,
Yeah, I mean, it's an old train wheel, right? Scientists, I feel like.
does anyone know if during one of the previous, do you know if during the previous expedition they dropped plates or something for colonization?
Oh no,
I
that's
don't
a
know.
weight.
Sieve. Sieve. With a wire runs on. It's from our gym.
Or a sail
Looks like a lion.
Okay.
Chain. Yeah, that's probably a fair call. Either acoustic or timed.
Thank you.
So we're just exploring this feature because it's at the kind of the highest elevation off the seafloor bottom from the site that we're sampling before we proceed on to the next waypoint. Did she say it was okay? Anna said it was okay? Yeah, okay. Okay, thanks, Marco. I think someone mentioned yesterday that we hadn't seen that many echinoderms in the area, that seep area, at least during the time that I was on watch. And then as soon as I left, I looked up at the monitor in the lab and saw a sea star, a few urchins, and so on. So if you were following along yesterday, we were definitely in some seep areas and saw very characteristic animals. associated with those seep areas. That's what we're looking for today, targeting an area like that so that we can collect some samples from those areas, like some of the mussels and my favorite, the sponges, some of the shrimp, gastropods, a couple crabs. We've already collected a couple corals, I think one stylaster and an octocoral. Of course, some of the tube worms. Okay, you ready to head? You want to head down that? Head down. Okay, and then you see. So we want to go. Okay, got it.
Okay, so you zoom into the field.
Yes, sir. Use vessel 500 meters east.
East.
So we want to come back down the ridge where we've just come up. See at the top. So east and up to 4.
Right. Yeah.
Somewhere right here.
Yeah, so we need to come.
This is the bridge here.
The track that we just done, we need to go back to the coast to the east.
So you like.
So I need to follow this.
Yeah, here.
You wanna come
So,
back
Matt,
here?
Claude, yeah, man,
But here was
Claude, right up there. She's pointing to up there. That, do that?
or you wanna go back there?
So maybe we'll go like this, up to far, like
Okay.
Yeah,
not exactly the same track.
yeah,
Something.
yep. Good. So in mission control, we have several people, all who have jobs to do, tasks to do. Of course, we have people who are crucial to
Hey,
operating the
you
ROV,
point.
our pilot and co-pilot. We have our multimedia technician who's handling a lot of handling this, I think this live stream and other communications. My job is dive lead for this hour. As much as I'd like to be able to narrate the whole dive and tell you stories about other interesting things, my job is really to be able to make sure we're following along the plan that our chief scientist, along with the others who are contributing to this, have put into developing this plan for this dive, and then communicating directly with the pilot about what it is that we want to do. And then also to be on the lookout, because we have targets, we have goals for each dive, and goals for this diver specifically to look for some interesting seep sites and collect the characteristic organisms that are there. And then in the second row we have our watch lead. So that's Anna. She's almost done. Okay, we're seeing bacterial mats now. And Anna was here for about, I guess, the first half of our 12-hour dive, and she was kind of overseeing things and making sure there's continuity from one dive lead to the next each hour, and making sure that we're sticking to the plan, helping us to make changes if necessary. And then we have other people in the room who sometimes I'll turn around and ask if they know what something is that I can't identify. We've got Jamie, who's she's logging observations of things that we see, like this crab, and taking notes, logging notes in a program that BIA has created, our next launch lead. So we're documenting everything that we're seeing and everything that we're collecting, really extensive notes. And then those will be publicly available as well. Okay. Waypoint 4 is where we were, where they were in 2014? Okay. All right. Thanks. All right. So we're heading to, so she doesn't want to, we're not going to go down. Just that's where we did. Okay. All right. All right. So again. Okay. So we're going to follow this ridge, head towards waypoint 4. Our chief scientist has reminded us, informed us that the dive that was made in 2014 is the site that we've selected as waypoint 4, and that's the one where there was confirmed seep organisms. So hang on, we're on our way. Those white streaks on the rock face are bacterial mats. Again, characteristic of methane seep areas.
Come north, like forward.
Huh?
Get the vessel to start coming north.
Come north now?
Yep, please.
Bridge, new waypoint.
Pilot and co-pilot are in close communication with the bridge, making sure that the ship remains in position, so it doesn't pull the ROV along. And so as we're moving along to our next waypoint, the ship is also moving along, repositioning as needed.
trouble?
No.
Currents again?
No.
Remember we have a long teller, so the longer the teller is, the delta is more risky in the red zone, so you really have to try not to be in.
Okay.
We're starting to see evidence of tube worms. This is a good hole at the bottom too. Oh, it's rugged, I know.
You want us to follow waypoint 4 along that line? You want us to follow the waypoint?
Yes. Please.
Number 1, 2.
No, it's good. I sort of come east, because they want us to come up the line.
You want to come on this line?
Yeah.
So you want your vessel more east?
Yes.
Mm-hmm. As you can see, it's pretty rugged terrain, so pilot and co-pilot are kind of flying up and down, getting around some of these larger boulders, trying to still stay on track to get from waypoint 3 to waypoint 4, which is where we expect to see more of the seep organisms. So we have a couple of columns that we have to clean. So if you see, get to an area where they're not like in a crevice and you think you're right. This large boulder is loaded with the stylasterid corals. As you can imagine, it's kind of challenging to navigate around these big boulders, rocks, making sure the umbilical doesn't get snagged on any of them. So we're seeing evidence here of tube worms and dead mollusk shells, some bacterial mats. Just looking into a crevice now. Just sure do look like it. There's some live mussels there. Don't worry, I'm not going to ask you to collect them now. Keep on moving towards that waypoint. Shells from dead mollusks. Starting to see some more octocorals. Let's keep going towards. I'd really like to get to waypoint 4. Yeah. We do have 2 species of crabs that we would like to collect as we get closer to the to waypoint 4, the area that was documented in 2014 as a methane seep area. Yesterday's terrain being quite as rugged as yeah.
Updated waypoint bridge. Our depth is 1,052 meters. We're heading to a target site that we identified, calling it Waypoint 4.
It's on this mound called Dome 2. It's the location at which seep was documented in 2014. On a tripod?
Want to catch it?
Yeah. So we're going to try and collect this crab.
Wait, you could come to a stop.
You can get some good video of it with and without the lasers.
All the lasers? Without.
Can we get video with and without the lasers, please? Yeah, you think he's going to start?
You can turn the lasers on. Okay.
His lasers are 10 centimeters apart.
I can see just below him on the rock.
Mm-hmm. Mm-hmm.
You wouldn't be able to stay here. You have to come back up. So you could put the current flowing into the headers. current is blowing you, rotate your head towards the current. You give up already?
See?
That's more challenge money.
Give up one chance.
Co-pilot's ready to give this a shot.
How's it going?
Wow.
You gotta rotate clockwise, anti-clockwise.
How do you feel now?
He was on this rock. That's the rock he was on.
Back and forth.
Okay. That crab got away. We're gonna keep heading towards waypoint 4.
Remember you sitting down, like those things on rocks and stuff. Always better put your head towards the current. It's just gonna blow the right side. So I can tell the current is by this fish. See that? Yeah, it's heading the current.
Again, more evidence of dead mollusks, dead tube worms.
I was waiting for you to start moving around it. Rich, you can continue with that.
formations, huh? Big slab there. So this is a 12-hour dive from launch to recovery, back on deck. And will still have about, let's see, it's almost 5 o'clock Eastern here. Probably have to leave on around 9. So we'll still have about 4 more hours to explore once we get to waypoint 4. Starting to see some of the sponges that are associated with these seep communities.
Kick it up. Yeah.
Excuse me. Vibrations are incredible. Gonna try and collect that crab?
Probably not on that cliff.
Nope. Okay, we're starting to see some like mussels now. Mhm.
Go.
So this hour has really flown by, and now we've gotten to waypoint 4, which was our destination, and I'm going to be passing the baton over to my colleague, Zahir Hussain, who's curator at the museum at the University of the West Indies, and he's going to be busy. He and the ROV crew are going to be busy for the next hour because we're at this site where I can see several of the target organisms that we'd like to sample. I'm just going to give him some notes about where we are, what we've been doing, so he can take over. And hope to see you tomorrow. Okay, so just good, which is the area
So the methane hydrate,
so we
so
don't
like
collect
just
anything
some
from
point
the
over
last
here, like make sure.
in my house. They've got both corals. They've got this Tylasso.
And they've got like while we're here, let's just get lots of like
this.
nice imagery.
So we need mussels. The sponges, I think, can be suctioned up. I think.
Is that a sponge underneath there, Shirley? Underneath the rock?
And it looks like it might be either an anemone or looks like it might be an anemone. Yeah, so we're gonna need the mussels.
Sponges that these guys.
Yeah, and I'd wait for a bigger clump and then should be able to suction that right up into.
Open muscles first.
Mussels. Yeah, we need mussels. Have fun.
Thank you.
Sorry.
Oh no,
Too many. We're gonna be busy
so, like,
tonight.
There's like zonation. These are like right by the flow, and then the tube worms and the sponges are like a little bit more out, and then the corals are further back.
Good afternoon, good evening, and good morning to our viewers. My name is Zahir, and I'll be taking you through the next hour of our dive. Currently, we've just come up on our waypoint number 4. We're going to settle and collect some of these very beautiful mussels that we see in front of us. We want to get about 7 of them. And we'll also try to collect some sponges, some of these white sponges you see over here. Hi guys, happy to be with you again. So the ROV is just gonna settle.
They think they're in. When you sit down, then we'll get like really good. But there's slope.
And then we're gonna try to pick up some of these mussels. We'll take about 7 of them. We'll hold 1 as a voucher specimen in the museum, and the other 6 will go to Dr. Kanhai, who's looking at heavy metals and microplastics. Bivalves are an interesting group of organisms to study, especially when you're looking at things like microplastics and heavy metals. They're filter feeders. And if you look very closely, you may actually see their siphons that they take in and let out water, filtering them with their bodies. So by studying the tissue of these mussels, we're able to see what type of chemicals, what type of heavy metals, possibly microplastics that are in the water and get trapped in their bodies. And we see a host of squat lobsters.
The sponges, surely.
Old specimens.
There. You're done. Like that's the.
So you want to go for the mussels first? Want to slope them?
First the slope.
Slope, yeah.
Thing?
Yeah, it's slip over there. For the mussels?
So like.
You think it'll work?
Only patch.
Scoop a bunch.
And then there's always half the ponds. There's just like loads happening in there.
Scoop will be done.
Pick it really soft, George.
They're suggesting taking it with the jaws instead of the slipper.
I don't know if it's gonna. It might be too big.
For the mussels?
Yeah.
Yeah, no, not for the mussels. For putting up like the streams and other.
Oh,
the shrimps.
Okay,
Oh,
so
yeah.
I guess we'll do a.
Streams, amphipods.
I guess we'll do a slope first to see what we get. So we're just deciding on what's the best method to take up these samples. I think we'll do a slurp first to see what kind of small crustaceans, amphipods that we can get. And then we'll probably go in with either the scoop or the arm to pick up individual or groups of mussels. It's a beautiful anemone, almost upside down there. And a host of squat lobsters, crabs, shrimp, crustaceans. A very, very diverse area here. And for those of us who were complaining about no relief, well, there you go. We've got underwater mounds, huge stones. So we're gonna try to slip up some of these crustaceans here. That's the suction arm coming out. You wanna go in one?
One is too big, so it's just for cleaning, so we can
Maybe
maybe.
two first.
Yes, please, and we can do 2 and 3.
Okay, I'm cleaning one. Yeah, okay.
So we might get some green ones with a bit of it.
Clean. Sure. So, we're going to, right?
Yes.
Yep.
Shrimps, right?
Shrimp, yep.
Like all the little things.
We're trying for, we got a squat lobster there.
There's limpets.
We got those already. Oh, we're trying to get those. Yeah.
Yeah, we were trying to, but not just the shrimps.
Yeah. Yeah, those shrimp are hanging on for dear life. But we'll also try to. So quite opportunistic right now with this slipper sampler. We're trying to get as many different types of organisms as we can, this being a very diverse spot. Another squat lobster. We may have got that one. And these will go straight into preservation. No chili sauce for these guys. And as the mussel gets disturbed, we see quite a host of crustaceans floating around as well. So it's quite diverse, very diverse area. We got one. I think we got one there. So I think we did get quite a few organisms. We're going to continue trying to get a few more of those shrimp. There's a few snails there as well. And the big crab just chilling. Another one. You may get a few here. Nice. These look to be paneaed shrimp. Paneaed shrimp are characteristically release their eggs into the water column after fertilization, whereas their cousins, the caridae, tend to hold on to their eggs until they hatch. We're getting quite a few now. Excellent work there by our ROV pilots. It is really tricky. The current is not very forgiving at all. And another one. Seen some limpets encrusting the mussels as well. They're sedentary, so things tend to grow on their surfaces. They don't move around.
So I think those might be easier to figure out the
Agreed.
You may not be able to see it, but we have a shot of the suction sampler, and there are quite a few organisms in number 2 already.
You got someone?
Let's try and get a couple more now, if we can. Some isoplods floating around as well. And
Super cool.
Exactly. It's like a level.
oop, there goes another one. I think we also picked up a little fish there. Yeah, we should be good. Come on. May even get a muscle. And hello to all our listeners from North America, Canada. Happy to have you all. Please keep those comments and questions coming in. It really does help our channel. It's a big guy there. Characteristic upward curved rostrum. It also seems to be barbed on the dorsal surface. We'll do some further examinations when they come up. But the rostrum is one of the distinguishing characteristics that we use to tell some of these species apart.
I think it's full already.
Mhm.
Maybe we can change, try to make sure we collect some of the gastropods. And there are some gastropods, we have like the different ones, some are really like without the lines, yeah. But I think it's better if we get some with the like with the whole muscle.
Okay.
There, I see some here. This one has some gastropods attached.
Yeah.
So I might get some of those.
One for the front. Yeah.
Yeah, no, those are eggs. But on your left, this one has a gastropod at the bottom. You see? It has a tiny gastropod, white one.
So maybe this one here, and
Yeah.
others like it would be.
That's it?
the little gastropod. Yeah, we should be all right with the shrimp. So we're going to try to take a couple mussels with the gastropods attached. Them being very sedentary organisms, all kinds of things grow on them.
Maybe when we get to the other place, we can move like another slab.
So we'll be changing tack a little bit.
What's next, Carl? Mussels next?
Yeah. But the ones with the, if we can, the ones with the little gastropods on them, the little white.
White. Okay.
Yeah.
I'll use the next one. Yeah. It's gonna be.
So
Yeah.
and number two is chock full of all kinds of organisms. So some of the white flecks that you're seeing are eggs, but some of the white flecks are also gastropods. So we're going to try to collect those that have some gastropods on them, because we'll be able to get two organisms for the price of one.
So where do you want these going? In a bio box or a quiver?
Bio box?
We might collect like at least
7 or 8.
I think more. So let's use a bio.
Bio, yeah. Well, we have all our bio boxes are open, so we can go with what's the easiest one for you?
Any of them?
Okay, BB3A.
Yeah.
Yeah. So we're going to maneuver and try to collect some of the mussels with gastropods attached. Okay, we'll be going in 4A. We'll try for 4A. one just slipped out. But we get a few more. Very delicate maneuvering by our ROV pilots. We're using the soft arm. Those yellow tabs that you see have actually been 3D printed right on board the vessel. And an amazing vessel it is. Thanks once again to the Schmidt Ocean Institute. And just a little background on our hosts. Their mission is to combine advanced science with state-of-the-art technology, as we are experiencing here, to achieve lasting results in ocean research, to catalyze sharing of information, and to communicate this knowledge to audiences around the world, as we are doing right now. Their goal is to foster a deeper understanding of our environment. With the scoop, yeah. Yeah. And our first mussel goes into the box. Guys, you all want to try the scoop instead? Yeah, I think we might get better success. Yeah, let's try the scoop. Please.
But it was just might
Break up some stuff. Yeah, we could go for it.
Scoop?
Yeah, scoop.
Those that are like in the vertical part
Column.
of the rock.
Yeah. And scoop up. Maybe scoop up. But I leave that up to you. So Schmidt Ocean Institute is a private, non-profit operating foundation established in March 2009 to advance oceanographic research, discovery, and knowledge, and catalyze sharing of information about the oceans. Their founders, Eric and Wendy Schmidt, established this institute in 2009 as a seagoing research facility operator. The institute is devoted to the inspirational vision of Eric and Wendy Schmidt and promote open source sharing of information to better understand our deep sea and our oceans. So we're just pulling out the scoop. And we're going to try to scoop up some of these guys to join their compatriot in Biobox 4A. So whilst we will be doing some experiments on some of these guys, a lot of them will eventually find their way to a home in Trinidad, either at the UE Zoology Museum or the IMA's Marine Museum. Here they will be identified, preserved, catalogued, and accessioned into our permanent collections. And then they will be accessible to all nationals of Trinidad and Tobago, and really anyone who wants to come and have a look. But particularly, they would be available to marine scientists who want to further the knowledge about these individual organisms and about the ecology of the deep. Ecology is how these and other organisms, that's a great catch there, about how these organisms and others interact with each other and with the environment. Very, very dense outcrop of mussels here. Whoa, we got a full pan. And you see many mussels coming up together. They attach to the rocks by structures known as byssal threads. These structures are extremely strong, which make them very difficult to remove from the substrate. If you've ever had a mussel or cooked a mussel, you would have encountered these byssal threads and you would have to take them out. It's actually under experimental review right now, trying to isolate these compounds that make these byssal threads so very strong. Oh, that's a great catch. That's a great catch. Probably had one of them going into BioBox 4C as well. We're gonna go and try for a little bit more. Because of the large concentration of these animals here, it's okay to take a few. We're always mindful of the densities and abundances of these organisms and are very careful not to disturb the very delicate ecology of those systems, of this system. And if you look closely at these mussels, you may even be able to pick up on, just had one fall off to the side, but there's also a gastropod attached to that one on the side.
You want more or you want something else?
Let's try for one more scoop, please.
Yeah.
So if you look very closely in this shot, you may be able to see both the inhalant and exhalant siphones that allow these organisms to both take in water loaded with nutrients and with organisms, and the exhalant siphon that allows them to get rid of the water as well. Recently, we did an experiment back at the UWI. Great catch there. We got a full box. We did an experiment back at the UWI during our open day. Yep. Crab. So we're gonna try for one of those crabs. maybe with the arm? Sorry?
You're gonna try and get it back to the retention.
Sorry,
good?
Oh, close up, yeah. Yeah, so we recently actually did an experiment with some green mussels, Perna viridis, and we put them in an aquarium and we threw in some algal water with high algal concentration, and within about an hour we went from totally green water to almost transparent. They're amazing filter feeders, and it's a good way to show how they actually clean our water. Perna viridis, however, is an invasive species in Trinidad. It has been supplanting our local brown mussel, Perna. Good job. So we're gonna try to get one of the hardest creatures to capture, our decapod. This interesting crab. He's already on guard. So we may use the arms to catch him. But I think he smells I think he smells a rat. Making a bid for escape. If we do get him, maybe 3A or 3B. Yeah. Keep him away from the mussels a little bit.
It has to go in.
The big one. Okay. 1A, right? So if we do get him, 1A. If we do get him. So quite a quite busy evening for us here. But the work doesn't stop after the ROV comes up. For most of us, the work just begins. Each individual animal will be photographed. A cryo sample will be taken, trying to utilize a brand new, relatively brand new cryopreservation technique, as well as genetic samples. Traditional genetic samples will also be taken, and we will be keeping, hopefully, some of them as voucher specimens that will go into our collections. So we're pulling out all the tools to try to capture this very elusive crustacean. And just to remind us how we're able to be here and to be with you, we have quite a few partners that have contributed to this expedition. Paramount of which is, of course, Schmidt Ocean Institute. So a big shout out to them again. But leading the charge locally is one of our more vibrant NGOs. It's known as Species. It's a non-profit NGO founded in Trinidad and Tobago by Trinidad and Tobago nationals. And it undertakes scientific research, education, and environmental advocacy. Other institutions contributing to our expedition: the University of the West Indies, St. Augustine, the Institute of Marine Affairs. We have a couple scientists on board and one of us IMA scientists back on land. Just a big shout out to Dr. Anjani Ganesh. Other institutions contributing to the expedition are the Harbor Branch Oceanographic Institute, Florida Atlantic University, USA, the University of Calgary, Dalhousie University, Canada, and the National Museum of Natural Sciences, Spain. Other supporting institutions are the Ocean Discovery League, USA, Pure Ocean, France, one of my favorites, the Explorers Club, USA, the Pew Charitable Trusts, USA, and the Anthony N. Sabga Awards for Caribbean Excellence. So a big shout out to all of these institutions, and thank you so much for your participation and your contributions to furthering our knowledge of Trinidad and Tobago's deep sea. So Annette has had a workout today trying to catch some of these crabs. But we're in a good position, so hopefully it works out. And the crab seems to be very inquisitive of the net, and he's coming closer. Yeah. Guys, yeah, this one seems to have some eggs. So, yeah, we don't want to take him one of the eggs.
What about this little guy down here?
Where's,
Yeah, we can try with that.
yeah, definitely try with that one. No, so the egg sac is on the abdomen.
I hope
Yeah.
not.
So we made an executive decision there. The one we were initially going at seems to be gravid, carrying eggs by her abdomen. So we're gonna reposition the arm and try to go for one that does not have eggs. These So it's one of the criteria that has to be met before we can take an organism. We would never take one that is actually reproducing, or in this case, gravid. So our team lead has just decided that we'll not go for that one and we'll go for this other one down here. So we're just repositioning the ram. The ram is what keeps the ROV in place because we're actually not sitting on the bottom right now. We're on the side of a seamount and of a pretty large rock. So the ram helps to keep the ROV in place. It's gonna be a little bit tricky based on where the arm is and based on where the ram is. But again, we got the best in the business. And he also seems to want to come with us, so come on down. He just slipped out to the side there. Yeah, it's a really tricky position on the side of this large rock. Remember, the ROV usually sits on the bottom, and it makes collections much easier. But trying to collect off the side of this rock is proving a bit challenging. So we're gonna reposition and try again.
Sit down without this.
Nancy grab it, yeah.
Oh yeah, wow, that's a lot of eggs. Wow, wow, wow. Yeah, let's get a clip. Wow.
Oh, that's a beautiful shot there. If you could just hold a minute so we can get some frame grabs. Wow, look at those eggs. Each one representing the future of this species. So there's no way we're gonna take this guy or this gal.
Wow.
An easy way to sex crabs is to look at their abdomen. Males tend to have a shape kind of like an obelisk, and females tend to have a bell-shaped curve on their abdomens. That's a great shot. So we'll just try to reposition. Side, yeah, I saw it. This seems to be a real hot spot for not only the shrimp and the mussels, but also the crabs. And that's a lovely anemone upside down there as well. So we're just going to reposition. We saw another guy climbing up on the outside of this rock. But if not, we'll keep our eyes peeled for more as we move along. The sponges, yeah? So we could explore there. Yeah? Okay. If we're repositioning, could we reposition somewhere right around here? Yeah, when you turn, right? But let's see if we can get the crowd.
What's our on deck time for this one?
10. Wait.
No worries.
I'll pick up my.
10. 10. 10. So we can leave bottom.
Perfect.
Sounds really
Yeah.
All right, for anyone just joining us, welcome. Our on deck time for this dive, you might have just heard, is going to be 10 o'clock local. So you're looking at a little over 4 hours still. We'll leave the bottom a bit before that because we gotta get back up, but there's still plenty to see. So welcome.
So we just reposition. As we reposition, I just wanted to share a little bit of information about our lead research organization, Species. Species' vision, a noble one, is to have healthy marine ecosystems that are sustainably managed to benefit the Caribbean. Now, in that simple statement, there's a lot to digest. Their goal is to have healthy seascapes, but at the same time, to promote sustainability in the management of these very unique seascapes. So those white sponges, we're also interested in those white sponges. But when you settle, when you get a good spot. And we see many mussel shells as well. So one of the criteria for mussel growth is adherence to hard, clean substrate. Oftentimes you see them developing on one another in close, large pockets because conditions are quite favorable in these little areas. We also see quite a few of the white sponges that we're hoping to collect on the side of this rock. But we're looking for a good spot to settle so we can try to access both the sponges and the crabs. So Species' aim is to sustainably manage our seascapes. It isn't an institution that is against extraction. On the contrary, its goal is to have sustainability in our deep and in our seascapes in general. It's led by Trinidad and Tobago scientists, conservationists, and communicators. Its director is our very own lead scientist on this expedition, Dr. Diva Amon. But she's ably supported by a host of other very passionate individuals. Dr. Michel Casabon-Manette, Dr. Anjani Ganesh, Mr. Ryan Manette, who is also on expedition with us today and has successfully had, I think, 3 deployments of his Doris system, breaking records on the way. Dr. Farnaz Solomon of the IMA, fish biologist, also on our cruise looking at ichthyoplankton, as we see that looks to be an interesting specimen right there. Crab, possible collection. We want to get this guy here if we could. Yeah. He's got a nice space. An associate member is Dr. Jason Alamu, the first. Miss Janice Germain is an associate member and project support officer. Mrs. Jamie Lee Luchin, associate member and one of our scientists on board the vessel. Mrs. Kelly Manette Camejo, associate member. Miss Jessica Sadler, associate member, as we see our capture, possible capture, hopeful capture. We're gonna get him, we're gonna get him. Come on, guys. He's retreating under the rock. Smart guy, isn't he? But it's a behavior we see also in our land crabs. If you've ever tried to catch a crab in the mangrove, they love to retreat down their holes. And this guy is no different. It's probably why he got to this size. Yeah. Could we...
The metals in the
Could we back up a little bit? Because we wanna get, yeah, this white stone right here. That's a pretty big one. That piece on the pretty large piece. So shifting gears a little bit, we're gonna try to take the sponge while the crabs settle, and we'll make another try for the crabs afterwards. So this guy here, right? Another associate member of species, Miss Charlotte Tom, who is on board our vessel. We did ship to shore. I did a ship to shore with Charlotte this morning. She was amazing. And all of these individuals have made this, contributed significantly to having this expedition in Trinidad. It was, I've heard some of the backstory, and it was not an easy thing to do. To get this type of vessel that's in demand all over the world to come to our small island is a feat in itself. So much kudos goes out to the species organization. Big up yourself. I see a funny anecdote in the chat. We possibly need a chicken leg on a string to catch the crab. That's how we catch crabs in Trinidad sometimes. We tie a piece of chicken onto a string and pull him up and use a fan cover to scoop him out. So we're just positioning, anchoring the ROV for stability as we just bump into a tube worm there as he disappears inside his shell. Yeah, that's what we're gonna try. Yeah, they know. Get some pics. All right. So guys, as we settle, we want to take some screen grabs. Off the background. So if you could pull back out the camera a little bit. But when you settle. Because just having a little shift change on the ROV pilots. Could we just pull the camera, pan the camera out so we can get both the sponge and the background in some pics, please? Lovely, lovely, yeah? Look at that, people. Very, very dense population of mussels. And their neighbors are our white sponge. Lots of tube worms as well. Look at that shot, beautiful. Host of mussels, crustaceans, squat lobsters, shrimp, crabs, you name it. Fish, they're there. Wow, look at those shrimp. Come on. So the camera is gonna focus. Beautiful. Look at that squat lobster. He's missing a claw. And when you look, you can see some of the mussels open, and you're actually able to see the inhalant and exhalant siphons that I was talking about earlier. And very rarely would you see any bare earth. Any bare substrate, they're gonna colonize almost immediately. And we're seeing gastropods as well as possible eggs on some of these mussels. Oh, that's a great shot. Probably get some screen grabs here as well. A little sea star, is it? Good. Yeah, we're good. If we could make it try for the sponge now. Maybe the arm? Sorry? Niskins. Okay. So we'll fire on Niskin first because it's a sponge. And then we'll probably use the arm and grab it. Put it in one of the bio boxes. So we're gonna fire what we call a Niskin bottle. This Niskin bottle collects a sample of water. Now, one of our researchers, we call him affectionately Sponge Rob, is doing some research on eDNA, but particularly interested in the water that sponges have filtered. So we're gonna fire a Niskin bottle to get some of this water so he can do analysis on said water for the eDNA of, well, any organism really that would have inhabited this area. Some of their DNA would most likely have been filtered. Yeah, only one. Which one was it? So we're just going to reposition a little bit to get the best angle to take the sponge sample. And one interesting question that popped up in the chat a few days ago was, many of the deep sea animals that we're seeing are actually red. How is that? Well, that's an interesting question. And things tend to be a particular color because they absorb all parts of the visible spectrum except the part corresponding to the color that you see. So in reality, because there is so very little or next to no light down at these depths, these organisms actually look black. So all of those red organisms would actually be black. But when we shine our light on them, the pigments in their skin actually absorb all parts of the visible spectrum except the red part of the spectrum. So that gets reflected, and hence those organisms look red when light shines on them. But remember, they live in an area where there is no light, so to most other organisms, they appear black. I found that a quite interesting question and observation by many of our viewers. One and three is occupied. Sponge.
Should be activated? No.
I don't want to put them too close to the muscles. Just making collection easier. But we could go in a box. We might need to put stuff in the quiver. Let's see what comes off. Yeah. So, are you going for the whole piece? No. If you could, yeah. But if it is the whole piece, it'll probably have. Okay, so could we just hold off 1 second? We've had a request to suction it actually. Could we do that? Yeah, let's try and suction it then. We need to probably pull back. As we see BioBox 4A chock full of mussels, we're just repositioning. We're going to try to use the suction sampler. So we're just trying to position ourselves to be able to get the best angle on the suction sampler. That'll go in. I think that one will go in number 4. Because we have something in 1 and 2 and 3 already.
Suction
some.
Mm-hmm.
And then we went to grab a bunch of
Yeah.
Just a piece. We'll want to take another piece, probably with the arm, and put it in a biobox.
Beautiful work there. So we just gotten our first sample of this white sponge with our suction sampler. It went into number 4.
So you take the biobox now. Not from this one, eh? Okay.
So the goal is to actually get multiple suctions, but from different ones, if we could. Yeah. If we could cast around. There's some over there as well.
The
Same species, yeah. No. We want a different one, but we also want to rinse in between. Yeah, sure. So this guy over here for suction, but that will go in suction 5. But we would like to rinse in between though. This guy here. Yeah. Success? Yeah. Maybe one more with the suction.
Then try number 6.
Yeah, 6. Yeah, we'll keep it separate. Maybe we can drop down a little bit. Perfect. So this one. So white branching sponge in 4, 5, and now we're going to try the 6. Perfect. We're on a roll here now, guys. So next we want to use the arm and put some samples in back. Yeah. There are pieces in there, so. So we didn't get it in 6, yeah?
6, yes.
Yes, we did. Okay, great. So yes, with the arms, we want to use another individual and put him in the bio box. Yeah, 1, 2, 3, yeah. Yeah, 7 and 8 empty, 1 to 1. So now we'll use the arms to collect another piece, but this will go in the bio box for a different type of analysis. You want to take maybe the top guy? But I leave which box you put him in other than 4A and 4, I guess 3A. Yeah. So we're going to try for 3A. Yep, and I reiterate, we know more about outer space than we actually do about our own deep sea. But have no fear, we haven't forgotten about the crab. The one on the top, yeah. This guy here, any piece of it, it's fine.
Yeah, no worries.
BB3A.
Yeah.
Right, I'll see you soon.
Okay, sounds good. Thanks, Jimbo.
With the touch of a surgeon, we've just taken our 4th sample of this white branching sponge. It's gonna go in bio box 3A. And this is in 3A, right? Oh, we got a hanger on. A little sea star. He's gonna come in the box too. Hopefully he stays on.
Is that 3A?
3A. Because we have stuff in 4 on the 4 side. So we'll go 3A. Or 3B, whichever is easier for you. I mean, it looks like everything is easy for you. That's a great drop. Thanks for that. And the box is closed. pull out and get some last screen grabs, yeah? And then let's search for our very elusive crab. Yeah. And rest assured, sponges are some of the most resilient organisms. They're going to be all right. They heal themselves quite rapidly, and in fact that is also under study currently. So this sponge is going to be just fine, and he seems to have a positive relationship with these sea stars. Great shot. Great shot. All right, thank you. We can pull out and possibly look for the crab. Let's catch one of those guys, man. Is it time? So my time with you has come to an end. Just want to say from the midnight zone to your screen, thanks for diving deep with the cucumbers, the crabs, and the mussels today. This is Zahir signing off from the deep sea where the pressure is high, but the thrill of discovery is even higher. Thank you. I'm going to pass on now to my colleague, Dr. Megan Conklin.
All right. Just as Zahir said, my name is Megan Conkling. I'm from Harbor Branch Oceanographic Institute from the United States, and we're going to proceed with collections.
Or you guys want to explore more?
I think let's just proceed, and then if we see anything on our target list, then we'll stop. Thank you. around up here. Going that way. Go forward? Okay. Yeah,
Just
we can
to
just go up that
start-
way. Just go up around this wall. And then we believe that that's where the hydrant is. So we're continuing to see a lot of seep organisms like mussels, clams, tube worms. Hmm? Okay.
Here.
They want to look below the ledge here, yeah. Can you swing back to the left?
I think I have a wall.
Yeah, if we can look under, like, around there maybe? No? Yeah, if we could just look underneath there. Perfect. We could try to collect that. If you guys are willing.
You want to collect the grab?
Yeah.
Yeah.
Why not?
What is this best?
Positioning, yeah.
Facing the beast.
Do we want to try to collect the crab or not? Yeah, I know.
You could try.
They can't really get in there because of the overhang, so I don't know.
You can't do that, right? Because it's not a computer. No, but she thought you were replacing. So you'd ask her if you were
Hmm?
replacing.
No, I'm okay.
So it was
I just don't think they're gonna be able to
She
get
wasn't
in.
doing anything. So here, well, that's what. So in order to avoid all these confusions, I have to ask.
But we'll just get, yeah, good shots of it after this attempt. Okay. We don't have to. We can just do some observations. They think that the yeah.
Yeah, alright.
This is the fun part.
Come on. The temptation is there. And he's got a nice little treat in his hand too.
No, no, it's not from the box.
Okay.
Yes.
I think so. We have both. We have, yes, we're both.
They're just too quick. They're just too fast. Yeah. He will not drop that treat.
So where the hydrants are? Sponge. But the idea is here.
I'm assuming they're up ahead. Like,
You can just
yeah.
zoom out to see.
Do you want to give up on the crab? Okay. We're gonna give up on the crab and look around.
We're not gonna
That's him.
We're not gonna catch a pause the crab.
Hunt.
For now.
Yeah. And we're just gonna pan up and look
Under
for.
the-
Yeah, that would be great. Thank you.
I don't think if that one fits in the net.
I'm assuming she's talking about here.
I'll
Yeah.
spread it.
Yeah. So up kind of. Yeah.
Yeah, getting a position in front of the.
Out.
Yeah. Rachel's screaming hydrate.
What?
Yeah, so into that area. So that white area in the center of the screen is a hydrate, so it's coming from potential seep area.
Someone will store in the net.
Okay, perfect. Everything is stowed away, so now we're gonna explore. I would like some zoomed in shots too of that, the underside of there. We have a lot of excitement from our lead hydrogeologist on board. She is very excited to see this. Do you want to take a temperature measurement?
Niskin? Sure.
Temperature?
I mean, I think we know it'll be cold.
I don't know.
Yeah, no, I mean, it's just, it's wild to see how much there is. It'll go on the sub.
Yeah.
But it'll all be gone by the time it gets to the surface.
All right, so we're just trying to get some good screen grabs.
Yeah.
Is it possible to pan the camera up a little? Okay. Yep. Yeah, that's what the plan is. Yep, we're gonna pan up.
No.
Whoa.
Well, it's a bigger zoom. You guys,
Sample
spot. Não conseguimos parar para mexer com a câmera. I want to see from bottom to top.
Yeah, if you could just do a quick zoom, that would be great.
After they've done with it. Yes.
That's a great position. Beautiful.
Right, and then just where they see you know this one. Yeah. Yeah. That way. It's been waiting.
Not there. Okay. Okay, so we're gonna try to get some zoom there and get a temperature reading.
Turn the lasers off. Look at the lights. It's very.
Is it struggling to zoom because, or the focus because of the, yeah, okay. All right, can we take it with the lasers off? Perfect. It's hard to tell, but it looks like it.
Okay, so
I can put unknown.
we were given permission.
Unknown.
We were given permission to use the light from them
Okay.
to, you know,
Eliminate
the
it.
arm where lighting stays.
Okay.
So we can do that first.
Can we use the light in the basket in order to illuminate this better? And we can use it with the hand.
No, we're looking at the pictures.
Let's try with the knife. Might be better.
Yeah. I just can't get it in focus because of all those.
Yeah.
Okay.
Oh, it's just in the shade.
Yeah.
Oh,
It's cool.
Yeah, they're getting the BBC Live. You can actually see the film. Yeah, it's also, there's a trap.
Yeah. Mm-hmm.
Handle? Yeah, I think it is. Yes.
Um,